हेलो। ठीक है। Hello. Shall we start now? Good afternoon, everyone. On behalf of Amity University, Uttar Pradesh, Noida, and the organizing committee of the International Conference on Sustainable Biotechnology and Bio-innovations, it gives us immense pleasure to welcome you all to the session of Environmental Biotechnology. We extend a very warm welcome to our esteemed chairs, Dr. V. Vivekanand ji and Dr. Doili Sanyal ji, whose contributions to environmental biotechnology continue to inspire research and innovation. We also warmly acknowledge our session in charge, Dr. Guru Murthy ji, for his guidance and support in shaping this session. A special welcome to our distinguished, invited, and plenary speakers, respected faculty members, researchers, industry experts, and our enthusiastic students joining us. Your presence truly enriches knowledge exchange and collaboration. This conference, organized by Amity University in association with the Biotech Research Society of India, International Bioprocessing Association, and the Center for Energy, Environment and Sustainability, with UNIBIC and IOT as gifting partners, stands as a testament to our shared commitment towards sustainable biotechnology and bio-innovations.
Today's program is designed to be both engaging and impactful, featuring insightful keynote lectures, thoughtful, provoking presentations, and interactive discussions. Here, we encourage all participants to please keep their talks clear, focused, and time bound to ensure an enriching experience for everyone present here. With that, let's begin today's proceeding. I now have the honor of inviting our session chairs, Dr. Vivekanand ji and Dr. Doyle Sanyal, to take a dice, please. Okay. So now I would like to introduce our first session chair. So now I would like to introduce our first session chair, Dr. V. Vivekanand ji. Dr. Vivekanand ji is a globally recognized researcher in environmental biotechnology and sustainable technology. He is currently serving as an assistant professor and associate dean at MNIT Jaipur. He is the PhD alumnus of IIT Roorkee and has been consistently featured in the world's top 2% scientists from 2021 to 2025. He is a recipient of prestigious fellowships including Ramalingam Re-Entry Fellowship and the National Doctoral Fellowship from AICTE. He has led multiple international collaborative projects across Europe and Asia in wastewater and biogas research. With extensive postdoctoral experience in Sweden and Norway and over 148 high-impact publications, Dr. Vivekanand brings exceptional academic leadership to this session. A big round of applause for this. So now I would like to introduce our next session chair, Dr. Doily Sanyal ji. Dr. Doily Sanyal serves as the Director of Organic Agriculture at Amity University, Noida. She is a distinguished academic leader with extensive experience in sustainable agriculture, organic farming systems, and eco-friendly crop production technologies. Her work focuses on promoting soil health, biofertilizers, integrated nutrient management, and sustainable agronomic practices aligned with the environmental conservation and food security goals. She continues to engage with national and international platforms to promote organic agriculture as a pathway towards sustainable development and resilient food system. So let's put our hand together for a big round of applause for our session chairs.
We would request the session chairs to kindly take their seats and lead the further proceedings and invite our eminent speakers. Welcome you.
Yeah, the first speaker is Dr. Suresh Kumar Dubey from Banaras Hindu University. He's going to talk on bacterial degradation of fipronil, a mechanistic insight. Dr. Suresh Kumar Dubey, you have, I think, 30 minutes to deliver. 20? Right. Fipronil. It's very close to my heart because I am a pesticide chemist. So let's see. I'm very inquisitive to know.
ठीक है सर वो आपके पास ये तो आ रहा है ना सर ये आप नेक्स्ट स्लाइड ये करेक्ट एक पॉइंटर तो पॉइंटर नहीं सर पॉइंटर करेंगे इंट्रोडक्शन चलाना इंट्रोडक्शन लाएगा इंट्रोडक्शन लाएगा introduction of these rights.
I hope someone is keeping the time, or am I supposed to keep the time? Organizers, please apprise me.
You will indicate, right?
टूट रहा है अभी बताए थे कि आपका पहले इंट्रो लगाना है
You can it would be nice having bio note first. Your bio.
You wanted to read? Wanted to read out?
You can read out and you can introduce.
Myself.
Or I
It's
can
not
do that.
necessary.
See, I
Uh,
can do it in a very nice manner if you want.
No,
Please.
just joking. In brief, good afternoon to all of you. It is very difficult to always introduce ourselves, but my name is Suresh Kumar Dubey. I am a professor in Department of Botany, Institute of Science, Banaras Hindu University, Varanasi. Today, I am going to talk about bacterial degradation of fipronil, a mechanistic bit की जरूरत नहीं है आप याद चाहे स्टूडेंट को सुनाएंगे नहीं सु देगा हम लोग 100 लोगों के क्लास में पढ़ाते हैं
चले
am I audible हाँ good कब देखोगे तब सुनाई देगा
ये इस माइक में बोल सकते
okay no problem don't worry I will see that so thank you chair for this permission and some known scientists and dear students and researcher. I am going to just share a case study done by my PhD student on this relevant issue, majorly on biodegradation, with the example of degradation of fipronil. So from this lecture, you can also plan your experiment, how to do the experiment at the laboratory level. So it is a graduate and postgraduate level experiment. Okay. More honestly. So the point is, at the outset, why we had selected fipronil. You know, a lot of pesticides have been investigated so far and their application as well. One of them is fipronil, which is chemically phenyl pyrrozole, and it falls under the, actually it's very difficult to manage the lecture without pointer. Okay. No, arrow is not going there. Not sell. है नहीं इस पे. Okay. So the point number 2 is, you can see that 7th most used insecticide in the world. So what I wanted to say that, okay, thank you. So what I wanted to show that, first of all, need to, before we start the experiment, need to think that why this experiment is essential. So a range of insecticides are available for the application. We had selected fipronil because it considered 7th most used insecticide in agriculture, industry and domestic sectors, and its half-life in laboratory varies from 122 to 128 and in soil 90 to 219 days. The chemically very dangerous because its half-life is too large. Another reason is, considering this effect against the insecticide, the pupils are using this insecticide in a huge amount, and that is why the WHO classified it class 2 moderately hazardous insecticide. So these are some points why the research on fipronil is essential or imperative. Okay, and this slide shows the since our air, water and soil are interconnected, so once contaminant come into the any segment of the ecosystem, it will transmit or distributed all component of the ecosystem. So here regarding one or two more points why the global community of scientists is worried about this. You can say that upon exposure, it creates a lot of problem neurologically, like some neurological disease. Up to this chemical have been detected in blood of the infants. Okay, and then the European nation also and United States categorized fipronil as carcinogen of the C group carcinogen. So lot of arguments we can give why the research on fipronil is essential. So considering this type of background, we try to study some literature survey. What is the knowledge gap about the fipronil related research, particularly for environmental concern? So we have found that some research data available, and mostly people try to isolate some bacteria and tested their potential for degradation of fipronil. But the very least information available, which gene or which protein or which enzymes are contributing to degradation of fipronil using bacterial potential. So, and few studies, largely most of the studies are utilizing some bacteria purchased by through culture collection center. So in microbiology, two concept is emerging. The one, the pupils are wanted to use bacteria, but they wanted to buy it from culture collection center. And some those who are working in the microbiology, they always prefer to isolate the bacteria from native environment and tested their potential. So we always encourage this kind of research to isolate the bacteria from native place and utilize it for your respective objectives. So considering that, we have designed several objectives like isolation of bacteria. I have seen several posters in the afternoon. Isolation of bacteria and tested their potential for fipronil degradation. Then most potent bacteria was used in a microcosm based study. Then we try to identify their genomic and proteomic basis for degradation of this toxic chemicals by using molecular docking or simulation dynamics approach. And finally, we did some metabolomics based approach also to complete this story. Here,
Yes, sir.
what happened?
सर जूम पे करना है सर 1 minute सर 1 minute
This is technical problem, not from my side.
नहीं सर 1 minute सर
So just for time adjustment.
yes sir
Okay. So for isolation purpose, these are the conventional approaches used by enrichment and biochemical, some batch culture and some GCMS based analysis. The students are regularly doing this laboratory in routine work, how to enrich the bacteria. There is no excitement about that. Here fipronil was used as a source of sole carbon and energy for this bacteria. So when we formulate the media, we use the MSM media, we have not used the carbon source except the fipronil. Right. Some biochemical test was done and all bacterial isolates were gram positive, gram negative both, and aerobic bacteria. Here based on 16 rRNA sequencing data, 10 types of isolates were detected and their genetic relatedness affinity based on the identified as Acetobacter, Streptomyces, Pseudomonas, Agrobacterium, then Pentoia, Bacillus. All are commonly growing bacteria into the agricultural soil. Okay, so our agricultural soil is also blessed with harboring this kind of useful bacteria. Okay, so now in next part of the study, once we have isolated 10 bacterial isolates based on agar plate methods, so we try to see the comparative potential of the bacteria for degradation of this toxic chemical. For that purpose, we have calculated the average relative fipronil degradation rate, and where you can say that the highest degradation rate was reported in fipronil, this FIP4 isolates, FIP4 isolates, and the maximum growth was detected at the level of 100 mg per liter. It means the bacteria show their maximum growth capability at the 100 mg per liter concentration of the fipronil. Okay, why I am emphasizing here? Because we have not processed all isolates for whole genome analysis. Because this experiment is very costly, so we have selected only one based on their average relative degradation capability of fipronil. Okay. Then after in next set of experiment, we try to see the growth pattern of the bacteria, where you can say if there is bacteria, it is expected that it must be follow the sigmoid pattern. Okay. So whenever we plan to work on bacteria related research, their growth plot must be essentially drawn. So in this cases, you can say all isolates show sigmoid curve, where the maximum growth was exhibited by this AS4 pseudomonas species, and simultaneously this bacteria have a highest degradation rate in this. So based on this data, we proceed only one isolate pseudomonas species. Here you look the comparative kinetic analysis. Here this one, this Km and Vmax and Ks ratio was found to be highest for this FIP4 was designated as pseudomonas, this one, pseudomonas bacteria. So this based on comparative this data, this bacteria exhibited more potential compared to others. Okay, so I have already said that several isolates were found positive for the degradation of fipronil. One of them was chosen for further analysis based on some kinetic parameters. So here we have tested, isolated and purified this bacteria based on batch culture study, vials based study. So you know that at least we will have to use the bacteria in field condition, in soil condition. So for that purpose, that experiment was designed in the form of microcosm study, where we have planned several permutation combination based study. In, for example, in one case there was a soil plus fipronil. It is known as natural attenuation condition. In another condition, fipronil plus soil plus bacterial inoculum. This condition is known as bio-augmentation. These are the several important technical microbiological terms used frequently in bioremediation based study. In another case, soil, fipronil plus nutrient, and this is technically known as biostimulation. And the fourth set, soil, fipronil, nutrient plus inoculum, it is combined of first and second. So in this way, we have designed microcosm study just only to say the performance of the bacteria which is purely cultivated in laboratory condition. Okay, to see the performance of bacteria in field condition, this microcosm study was designed. Okay, in this condition, in all the cases, in this case, the maximum degradation potential was observed in fourth condition. And it is obvious because this condition is more supportive for the growth and multiplication of bacteria, because there is no any limitation of nutrient. Inoculum is available, plus already inoculum is present in the fresh soil. So added inoculum is there. So that is why this condition is more suitable for the growth and multiplication of the bacteria. We had applied monad kinetics for the calculation of the rate of degradation in microcosm studies, and this equation was well fitted, monad degradation kinetics, for this purpose. And here, due to time limitation, here I was saying that this fourth condition was more favorable. It is based on some of our data calculated during the microcosm study. And then we found that up near about 86% degradation was reported in microcosm study, and around 85% degradation was reported when we use pure culture of bacteria in vials based experiment. Okay. So after that, in next objective, we wanted to see the which kind of gene and protein involved that enables bacteria to degradation of this toxic compound. So whole genome was done using some straightforward methods, and this you can say here, this more importantly, this GC, too much this GC content, around 65% GC content, suggesting the stability of the bacteria in toxic condition. So higher the GC condition will provide the higher the stability in the stress condition. Here, after that, we isolated the whole protein and SDS page was run. This SDS page only suggests that compared to control, the band was dense. So it just only suggests that some overexpression of extra proteins that involve in this process. Okay. This analysis was done using high resolution accurate mass spectroscopy. Our university is very rich in facility wise. So we are blessed with this kind of facilities. And then we calculate the intensity versus mass and charge ratio just to show any differences in peptides compared to the control. And when in the first half you can say the treated sample show some clumsy peaks where that is well segregated in the intensity versus MZ ratio. It just indicates the differences in presence of peptides in control as well as the treated samples. Here, this figure is important because based on some known information available for degradation of fipronil, we have identified 4-hydroxyphenylpyruvate deoxygenase enzyme, homogenizatase 1,2-deoxygenase, oxoacetate decarboxylase, and econitate dehydrogenase. This enzymes is reported to in degradation of this fipronil, and in our sample bacterial protein all enzymes were available in a appropriate proportions. So our the laboratory based incubation experiments suggesting the degradation potential was because of presence of these enzymes. Okay, then put this here. Everybody knows Ramchandran plot the significance. This plot is suggested the stability of the model structure because all the residues, the amino acids, falls on the suggested, the 2 and 3rd quadrant. It in all cases of enzymes, it suggested that the model structure are stable. Okay, then with the help of molecular docking, we also seen that the substrate enzyme interaction is stable in all cases which enzymes we have reported. In all cases, enzyme substrate interaction is very intense. Here some physical parameters also have been suggested like a number of hydrogen bonding or RMSF or radius of gyration or RMS. These are the physical indicators that pupils commonly used to just to see the stability of enzyme substrate reaction, and this experiment was done up to the 100 nanosecond, and up to the 100 nanosecond these variation was up to the suggested to the value, and this is indicate that the during this interaction between enzyme and substrate, the interaction was up to the mark. Okay, and in the final set of experiment, we did the metabolomics. Using the HRMS, we identify the important metabolites which after the bacterial, when the bacteria react with the fipronil. So we have found this, the important metabolites, the name is too big, like the first one is the fipronil pure, the second was the 5-amino-1, the second one is the dichlorotriphenyl, the third one is the hydroxycarboxylic acid, and the fifth one is the pyrozole, and the last one is the carboxylic acid. So these are the prominent metabolites we have detected in the elutes of our samples, and based on the available metabolites, we try to propose a conceptual pathway. Here you can say the fipronil, first of all it broken down into the fipronil sulfonate with the help of this cytochrome P450. Again it is broken into the 5-amino-1, then again into the fourth, it broken into the 5-amino-1,4 trimethylene, Then in the next case, it is in the 5-amino-phenyl. Then again it broken into the oxaloacetate, aminonatate, and oxaloacetate decarboxylatases. So this, based on our own laboratory outcome, we have proposed a conceptual pathway for the degradation of this fipronil by Pseudomonas species. Then the major conclusion is we have found Pseudomonas FIP-A4 is a highly potent bacteria isolated from the contaminated soil nearby the Varanasi, and we had validated the incubation experiment by genome, proteome, and metabolome insights, and a conceptual pathway has also been proposed in this regard. And as far as further suggestion is concerned, the laboratory which are involved in application of bacterial potential for decontamination of the contaminated soil can be used this bacteria because we have submitted all data to respective platforms. The culture was also submitted to MTCC Chandigarh. Then all sequences, right from the genome sequences or protein sequences, everything is deposited into database along with the culture of the bacteria. So those who are interested in this bacteria, they can ask from the MTCC Chandigarh, and they can search this genome proteome data through NCBI. This outcome published in ESPR, then chemosphere, then environmental pollution, and many more. And the credit goes to this student. She recently finished this work as a doctoral work, and currently my this very small laboratory warriors, you can say, they are doing extensively with the different works. So with this information, thank you very much for giving opportunities. I am right time or
You are even before.
yes? The teacher is always before finish the lecture, so there is no instruction to write. Okay. Yes.
Thank you, Dr.
We know
Dubey.
how shorten the lecture and how strengthen the lectures. So I would like if you have any query.
If anyone is interested in asking few questions, it's open right now.
Yes, sir. I have a couple of questions.
Yeah.
Excellent work. So my compliments. Because this fipronil molecule, it has chlorine and fluorine both, right? So definitely you need a dechlorination and defluorination for detoxification or bioremediation. I would like to know whether, because you have done the metabolomics also, so this, any of the metabolite, whether it was more toxic than the parent molecule during bioremediation.
Okay.
So that's my question. And yeah, only this one only.
Okay.
Yeah, thank you.
Right. This is very good question. Many times we debating this issue with some industry people, similar kind of questions. The most important study before releasing any chemical for the public use is analysis of metabolomes or metabolites, and which is very dark part of our industrial sector. Sorry to say, they are releasing the pesticide, but they are very hesitant doing the life cycle analysis of the chemical. Because once a chemical release into the environment, n number of metabolites will be there. I can quote a very specific example, then I will respond for that. Chlorpyrifos, very famous organophosphate chemicals, the domestic sector, agriculture sector, using it to kill the termites. Once you apply into the environment, its first hydrolyzing product is 2,3-pyridinol. So the parent compound half-life is 60 days to 70 days or 80 days. But TCP half-life is 150 days. And this first hydrolyzing product is antimicrobial and more toxic than parent compound. It is specific because this study I have completed in my laboratory and we have published several data upon that. So the metabolites in several cases are more dangerous than parent compounds. As far as this specific question is concerned, I have not done this experiment for fipronil so far, but in future this is my next plan.
Can I answer that? Sulfone and sulfoxides are more toxic than the parent compound, but since the metabolites you saw has degraded to the smaller fraction, it was not persistent, I'm sure. The sulfone and sulfoxide both are more potent than fipronil.
Because when they started working on DDT and it got biodegraded and they got a metabolite DDE, DDE is more toxic than DDT. Similarly, tetrachloroethylene and trichloroethylene, which are the industrial pollutant, groundwater, you'll find this PCE and TCE. After biodegradation, we are getting vinyl chloride, and vinyl chloride is more toxic than your PCE and TCE.
In several
That is the reason-
cases it is true.
And also the Pseudomonas, whether it's pathogenic or
No, it is non-pathogenic.
not pathogenic, right?
It is non-pathogenic.
But how come the species is not identified? You have done 16S RNA? The 16S RNA has been done, so I was inquisitive, which is the species then? You must have identified?
Yes, I have done the whole genome analysis.
Yeah, that's why. What is the species?
No, species level detection was not done.
But I think
It is what
We have
not
done
showing,
six-
not, it is
Huh.
what not showing a similarity with other reported species. And
Okay.
that is why we have
Okay.
written Pseudomonas A4P1.
Right.
I fully agree with her.
Right. We are not in hurry to say that it is novel.
Was
Unless
it-
because we are several experimental check is going on. Unless confirmation from all the side, we will not say.
So what is, was it fluorescing? Was it fluorescing? Did you do the fluorescence test?
No, no.
Pseudomonas fluorescence is very common.
We have not done this fluorescence based experiment.
And 16S RNA sequence homology, after blast search, how much you got it? Sequence homology with the 16S.
16S homology was around 92-93%.
So then I think it's a novel only. That you can consider novel.
That is why I am saying that something is going
You
on the
will,
way. That is why
you'll do further research.
Yes.
Yeah. So which HRMS did you use? Which detector and mass analyzer did you use? HRMS?
I do not know about some in-depth instrumentation details. This facility is available in BHU as a central facility.
Right. So who identified the metabolites? No, who identified? Means the structure, who gave the structures?
Technical person, technician will give.
That means you used the
They have-
database.
Yes.
You
they have
used
the
library.
They
Right.
have the library.
So these are all known metabolites.
Yes, yes.
And regarding the species level, if you want to identify that, I can suggest you can go for FEM analysis, fatty acid methyl ester profile, FEM. FEM, because 16S RNA probably it will give you up to genus level, but FEM, if you are doing it, then you can go up to species level also. So you can go for FEM.
This is going on. This is going on.
Question again. Now then I'll ask question for you guys. Thank you, sir. Thank you so much. It was wonderful lecture, and there is a lot of level work, applied work, patentable work. Many things are there. And since you are here and with such a, high in profile since many years. So my request to you is, since there are a lot of UG and PG students, I can guess, can you please give some tips to them? There must be some budding researchers here in the group, UG and PG, other than scientific technical part we have ended here, but just guide them with a few lines so that they can, who are thinking to excel their career in the science and research, they can follow you.
Right. So at this stage, I only can say that if you are interested to extend your career in academics, you must try to do literature survey very honest way. Because which always escape. The student always escape to do the literature survey. They try to do the literature survey within week or 1 or 2 days. So don't become hurry to do the literature survey. You identify the important journals in your related area. Do the literature survey. Suppose if you will search one paper in 1 week, it means in 1 month you will take 4 papers. And if you are here for 4-year BTech course, then you can simply calculate how many papers you have. And without literature survey, you cannot identify the knowledge gap. And without knowledge gap, you cannot set the hypothesis. And without hypothesis, you cannot design your objective. And you can simply say that you cannot do the experiment. So do not become the tool. You should not become totally dependent on your supervisor. Supervisor have many works. You search the papers, then discuss with the supervisor. And many times you do it in a reverse mode. Supervisor tell you, do the literature survey, then you come. We expecting that you do the literature survey first. In many interview, many times we are in the interview. Just after lecture, I shall have to move to the airport, and tomorrow afternoon I will be in Bhubaneswar just for a selection committee meeting. You can imagine simply in today's poster, I was asking a student why it is heavy metal. She was doing the degradation of heavy metal. I simply asked a basic question, why the term heavy metal? You can simply imagine. It is not a very tough question. You have always ignored doing the research on degradation of heavy metal, but you are not aware what is the definition of heavy metal. Simply. You have done lot of research. In an interview board, expert asked, you tell me a paper, journal published from India, have high reputation. We are asking that you say about the current science. Freely available and interdisciplinary. So the current generation is blessed with the mobile phone and internet media. They can get the information from the everywhere. But dear students, you do the literature survey very seriously. Otherwise, very hard to do the research, because the world is moving towards very competitive mode. You might be heard or facing, your supervisor is facing open access journal challenge. How can you pay this much amount? The funding agency is not generous to provide the fund.
Thank you, sir. I would like to add something, and for the students also. When you are going for a PhD, first of all, the sense of responsibility comes only and only when you find the objectives and the problems. When I joined IIT Roorkee for my PhD work, my supervisor told, कि बेटा तुम रुड़की में जाओ, what is the biggest problem near IIT Roorkee campus, find out the problem. And then I told them that Parthenium is the biggest problem. And the urban people are facing difficulty due to the Parthenium grass, Congress grass, and then the solution given by my supervisor. So when I started my PhD at that moment at IIT Roorkee, then the sense of responsibility towards my topic, always belongingness comes. So that's why, as sir told, if you find and search the problem and try to connect or discuss with your supervisor for making a solution, I think it will be more and more beneficial for your career also. Otherwise, supervisor has given, you have no interest about this topic and you take this PhD work as a burden. That should not be there. In continuation of sir, जैसा sir ने बोला, मैंने उसमें add किया है. Thank you, sir. Thank you. Thank you for it.
Finally, I would like to mention one point. Your teacher, Dr. Raja, was my student in UG and PG from Banaras Hindu University.
And good thing is that Dr. Ajay is the biggest scientist of MIT University of Biotech, MIT Institute of Biotechnology. He had published more than 300 research papers in Scopus Industrial Journal also. So one clap for Ajay sir also. And he is a student of professor from BHU. So you can understand the level of the professor who came today for a presentation. Circle we clap.
Yeah.
Thank you.
Thank you. Thank you so much, sir, for the wonderful lecture. Thank you. Okay.
Thank you.
Our next speaker is Dr. Bikram Basak from Hanyang University, South Korea. And Dr. Basak will be talking on enhancing methane recovery in electrochemical systems by using light. Dr. Basak research area is environmental microbiology, microbial diversity, bioremediation, and bioenergy via anaerobic coagulation. He has been ranked among world top 2% scientists since 2022 to 25 and has been conferred with Handitsa Award from South Korea and serving as associate editor as well as editorial and Springer. Dr. Basak, are you online?
Hello?
Yes, can you hear
Yeah.
us?
Yes. Yes, I can. Can you hear me?
Yes, very nice. Very welcome.
Okay. Yeah, thank you. Yeah. Hello, everyone. So, greetings from South Korea.
Yeah, they're trying to get the video as well as the slides. Yes,
Now it's okay.
we are with
Yeah,
you.
I can. I can share my screen.
Yeah, please share and start. Welcome.
Yeah. So, good afternoon, everyone. First, should I start? Hello?
Yes, sir.
Yeah. Okay. So, good afternoon, everyone. First, I would like to thank the conference organizers for giving me the opportunity to present my work here today. The title of my presentation is Boosting Methane Recovery in Microbial Electrochemical System by using food waste fed analyte. My name is Bikram Basak, and I'm currently working as an associate professor at the Resource Development Research Institute of Hanyang University in Seoul, South Korea. So in this presentation, I will walk you through a novel operation of anode chamber in microbial electrochemical system and how it facilitates the improved electron transfer and resulting impact on the overall methane recovery.
Dr. Basak, can I
Yeah,
interrupt?
yes.
3 seconds.
Yes, sure.
15 minutes, please. 15 minutes.
Hello. I couldn't hear your voice. Hello.
15 minutes.
Yeah, okay, right. So biogas is inherently variable. It typically contains 35 to 45% carbon dioxide along with trace hydrogen sulfide and other impurities, which limits the direct use of biogas from the anaerobic digestion, and which limits the direct use for vehicle fuel, grid injection, and high efficiency power systems. So conventional upgrading relies on the water scrubbing, chemical adsorption, membrane separation, etc. These are mature process, but often are energy intensive, chemically demanding, as well as capital heavy, capital expenditure heavy, and especially at distributed scales. So what MES brings to the table is that MES offers an alternative using electrode-driven microbial communities to reduce carbon dioxide into methane. So this biologically upgrades the calorific value of biogas while potentially operating at a lower pressure with a modular, scalable hardware. So first, unlike the conventional upgrading that removes carbon dioxide, microbial electrochemical system converts carbon dioxide into additional methane. So this increases the volumetric methane yield, pushing biogas towards the pipeline or vehicle grade specifications. Second, MES enables integrated waste valorization. So electrons can be supplied from the renewable electricity or from anodic oxidation of organic waste, allowing the circular coupling of anaerobic digestion and MES. So in practice, this lowers the thermal and water demand in MES and reduces the chemical usage, and it lends itself to modular on-site retrofits around the digesters. So this schematics here shows a dual chamber cell separated by a membrane. So electrons flow from the anode, that is the left hand side here, to the cathode, and proton traverse the separator, which is the proton exchange membrane. You can see here in the dotted line. So carbon dioxide is fed into the cathode, in the headspace or in the liquid, and at the cathode, methanogenic microbes accept reducing equivalents to methane and hydrogen and water. So the electromethanogenesis basis of this process is then there are two principal routes. One is direct electromethanogenesis, where carbon dioxide is directly reduced into methane in the presence of proton and electrons. Protons come from the anode, and electrons come from the proton comes from the anode through the proton exchange membrane, and electrons coming from the power source. And here in the cartoon, you can see at the bottom, it emphasizes the division of labor within the biofilm. There are many electroactive bacteria, hydrogen producing microbes, homoacetogens, and methanogens arranged, and they form the biofilm or the cluster near the cathode. And this special organization supports both direct and mediated electron transfer, which facilitates the methanogenesis. And in indirect, there is another methanogenesis route, which is indirect pathway, where carbon dioxide is first converted into acetate through the homoacetogen bacteria, and this acetate is then again converted into methane using the acetoplastic methanogens. So the conventional MES is often electron starved at the cathode. The conventional MES, which involves the water electrolysis at the anode. The limited anode electron supply caps the methane productivity in the cathode and pushes the energy demand up, because of more of the reduction relies on the power intensive electrolysis. So in this experiment, we set out to develop a modified MES, where we fermented the food waste in the anode. The goal was to generate additional reducing equivalents, that is the proton and volatile fatty acids, and the shuttle protons, and the objective was to shuttle the protons across the membrane to boost electron availability for the cathodic reactions, that is the cathodic methanogenesis in the cathode. So you can see the top schematic is the conventional MES, which involves the traditional water electrolysis, and the modified one involved the fermentation of food waste, which delivered the proton and electron to the cathode for enhanced methanogenesis. So this figure shows the cumulative methane production profile. That is the top one, and the bottom one shows the carbon dioxide utilization profile in the modified MES, that is microbial electrochemical system. So the modified system consistently, you can see, outperform the control, which is the blue, represented by the blue squares, and the modified one represented by the red circles. So what drives the gain? Actually, the dual electron supply. The electrolysis plus food waste oxidation increases the reducing equivalents, reaching the cathodes. So the strong and also the stronger biofilms, as we will see later, how the stronger biofilm, the modified configuration promotes denser and more active biocathode communities, improving the interfacial electron transfer from the anode to the cathode. And also the syntropy existing between the acetogenic and methanogenic partners enabled the acetate and acetate to methane route and also carbon dioxide to methane route, keeping the system productive as conditions may fluctuated. So the electrochemical readouts we received from the potential stat readings, the readouts reflects the gas data and explained why MMES performed better. So within each batch, you can see the current decline as the current declined at the substrate and carbon dioxide are consumed and then recovered after replenishment at the beginning of each batch. So this is evidence of a responsive, stable biofilm electrode system. Across the batches, redox peaks grow larger in both systems, which is represented by figure B and C, you can see here, and indicating the biofilm maturation and improve electroactivity. So critically, MMES system shows more pronounced oxidation reduction peaks, as you can see in the figure B. Figure B. And it is consistent with greater catalytic capacity and faster electron transfer at the biocathode. Additional electron supply from the food waste fermentation at anode increases the cathodic reducing power. So denser and more active biofilms in the modified microbial electrochemical system facilitated both direct electron transfer and hydrogen mediated route. To explain the electrochemical behavior further, we looked directly at the electrodes using field emission scanning electron microscopy and confocal laser scanning microscopy. So you can see on the panel A represents the MMES cathode. We see a dense multilayered biofilm with extensive surface coverage, and you can see, notice the rod-shaped and filamentous morphology of the bacteria. And it's a classical mixture of electroactive and syntropic consortia. And on the panel B, you can see the MES cathode. The coverage is there, but it's somewhat patchier, with smoother areas of exposed carbon filaments of the electrode. And it is consistent with comparatively lower current and fewer active sites. On the panel C, you can see the MMES anode surface, which is also colonized, and it reflects the active fermentation of food waste, which supplied additional reducing equivalence to the circuit. On the panel D and F, you can see the CLSM picture, which shows the live biomass, more live biomass with green, thicker, and more continuous structure across the field and through the Z stack on the figure F. These structural features map directly onto what we observed with the gas data. Higher sustained currents, stronger CV peaks, and faster carbon dioxide drawdown, and higher methane yield in the MMES. You can see on the panel figure E and G, the green, that is the live biomass, is actually lower than the modified one. So this reflects the liveliness of the electrode biofilm and the modified version of the microbial electrochemical cell. To link the microbial electrochemical roles, we also quantified the alpha and beta diversity for the planktonic cells and the electrode biofilms in both systems. And in the cathode planktonic cell, both OTU numbers and Shannon index drop from day 6 to day 19. A classic enrichment for the carbon dioxide adapted electromethanogenic consortia, because when the carbon dioxide adapted microorganisms enrich, the diversity become lowered. The electrode biofilms showed a lower richness than the early planktonic communities, but similar Shannon values, fewer taxa, and more even distribution, indicating selection for the electrode adapted specialists bacteria here. And if we look into the beta diversity, that is the principal coordinate plots, panel B and C. It shows a strong separation between the cathode and anode communities and a clear split between the planktonic and biofilm communities. So substrate regime and the electrode attachment are primary community drivers here. And MMES anode and cathode biofilms cluster tightly, suggesting a more coherently, functionally streamlined consortium supporting the electron flow. So the experimental setup promotes the propagation of electroactive and syntropic partners on the electrodes, which enhanced both the direct and mediated electron transfer. Resulting in the carbon dioxide to methane and acetate to methane conversion. So the community convergence on the electron aligns with the higher currents and methane yields that we observed. When we delve into the more details of the microbial community, we saw that MMES favors enrichment of electroactive and syntropic bacteria along with the key methanogens, enhancing the cooperative electron transfer pathway. As you can see, that is highlighted here in this by this red box. The dominance of methanoseta in the MMES cathode biofilms aligns with the higher methane yields via the acetoclastic methanogenesis supported by the diet. Hello? Yes?
Yeah, you have only 2 minutes left.
Yeah, I'm almost done. I'm just covering it up now. Yeah, thank you. So when we look into the phylum level across all the samples, Bacteroidata dominate, followed by Firmicutes and Actinobacteroidata, typical degraders and fermenters that feed downstream syntropy, which are present in the anode electrode. Archaeal phyla, Halobacterota, are noticeably higher on the cathode biofilms and then in the planktonic fraction, consistent with the electrode-associated methanogenesis driven by the methanoseta. And in the genus level, we can see the cathode biofilms are enriched with electroactive and syntropic taxa. For example, Geobacter, Smithella, Clochimonas. These are key syntrops channeling the organics to the acetate and hydrogen. And Geobacter supports direct interspecies electron transfer to methanogens. So all these species are associated with the methanogens and drive the carbon dioxide to methane conversion. So this division of labor underpins both direct electron transfer from the cathode and direct interspecies electron transfer and acetate mediated routes. So all these routes contribute to the enhanced methane production in the modified MES. So methanoseta is the most dominant methanogens on the cathode, indicating the acetoclastic methanogenesis as the primary pathway, but also hydrogenotrophs, that is contributed by Methanobacterium and Methanolinia, are also present. So they contribute about 20 to 30% of the methanes produced in the MMES reactor. So to conclude, our system achieved approximately 1.6 fold higher methane production from carbon dioxide compared to the control. And this enhancement was mechanistically driven by increased electron transfer and electron availability from the anode, from the food waste fermentation at anode, combined with the synergistic effect of dual electron sources, that is the electrolysis and organic decomposition in the modified MES. So electrochemically, we observed the enhanced electrochemical activity reflected in the higher current densities and stronger redox potential, as evidenced in the potentiostat data. And biologically, this translated into superior electrogenic biofilm formation, which was evidenced from the microscopic data with denser and more viable electrode colonization. So together, this result indicate that both the electrochemical and microbial frameworks both were strengthened and contributed towards the higher methane production. And metabolically, acetoclastic methanogenesis dominated via syntropic interaction, though hydrogenotropic Methanolinia and Methanobacteria also contributed to the higher methane production. So beyond performance improvement, this work demonstrated the practical integration of waste valorization and carbon dioxide utilization. So this carbon dioxide we supplied as pure carbon dioxide, but in the real practice, this carbon dioxide can also come from the 30% of the biogas. That is, 30% of the 30 to 40% of the biogas carbon dioxide can be converted into the methane, which can essentially upgrade the biogas to the biomethane level, that is the pipeline or grid level purities. So with further optimization and scaling, so this strategy holds strong potential for biogas upgradation, integrated waste treatment system, and renewable energy applications. So that's all. I conclude my presentation, and I'll be happy to take any question that you may have. Thank you so much.
Thank you, Dr. Boshak. Are you able to hear me, Dr. Boshak?
Yeah. I can hear.
I just have one question. So, what's the purity of the gas at the end of the day? Did you find out the impurities present?
We actually supplied 99% carbon dioxide, not actual biogas. And we received around 70% of that carbon dioxide converted into methane. We did not analyze for other impurities.
Yeah, because that's the important aspect when it goes for commercialization, that purity of the gas is important, right?
Yeah, purity of the gas is important, but in this experiment, we just with this prototype, we just checked the convertibility of the carbon dioxide to the methane.
So I'm sure we do further research to find out the impurities and how pure it is, and commercialization potential, right?
Yes.
Right.
And the impurities that may come is actually from the biogas, the hydrogen sulfide, siloxane, and other impurities that it may come from biogas. So when we feed the biogas, direct biogas into the system, then we have planned to check the impurities, because the biogas has to be upgraded or purified. And in this time, we just used 99% carbon dioxide to check the convertibility of this carbon dioxide into methane. I mean, how much carbon dioxide can be converted into methane, and we checked that it was around 70 to 80%.
Yeah, because the impurities can be very corrosive, like hydrogen.
Yeah, of course, you are right.
Holds importance, right? Thank you so much. Anyone else would like to ask any question? Yeah, we have one question for you.
Yeah, sure.
Thank you very much for such a nice, wonderful presentation. I have a couple of questions.
Yes.
First question is that you took PEM, proton exchange membrane, and
Yes.
anode you used analyte. So in analyte, there are more number of cation than proton. So it is expected that it will percolate through the membrane and it will make the catholyte more alkaline. So that may goes against methane production.
No, we used proton exchange membrane because it will specifically only allow the protons to pass through the membrane, not other cations.
Okay, that is probably Naphion, right?
Yes.
Yeah, but Naphion can also allow the other cations also. Okay, my next question.
No. It was proton exchange membrane. Proton exchange membrane. The membrane that we used, it only allows the proton to pass through the membrane, not other cations.
Okay, so then my next question is that you used the term called electroactive bacteria, but in when we used a biofilm, it calls electrotrophs. So do you think that these bacteria that you told, that methanogenic and other bacteria which is producing methane, it is a electrotrophs or electroactive bacteria?
I think they are electroactive bacteria. The methanogens are not usually very. They are not capable. Recently there are reports which some methanogens can accept electrons from the cathode directly, but usually these are electroactive bacteria. For example, Geobacter or Pseudomonas aeruginosa. They can actually extract the electron from the cathode directly. So they are electroactive bacteria, not the electrotrophs.
Okay. Thank you.
Thank you, Dr. Bashak. Let's have a round of applause for him.
Thank you so much for inviting me for this lecture, and I hope I have been able to explain my research well. Thank you so much.
Certainly, certainly. Right. Thank you.
Thank you so much, sir. Thank you for the insightful and thought-provoking interaction and presentation. And as we have Dr. Suresh Kumar, sir, he is very tight on his schedule for the evening. So we would just take small time, and I would be liking to invite the session chairs to felicitate Dr. Suresh Kumar Dubey, sir. Ma'am, answer, please. Can we have gifting partner? Thank you so much, ma'am. Thank you so much, ma'am. Thank you so much, sir. Thank you.
No, no. बोल दो. Okay, sir. Now I invite Dr. Mukesh Kumar Avasthi. He's a Marie Curie Fellow also, U Horizon 2020 contributor, and he does research on waste to resource bioengineering. Of course, I will not go on to how many undergrads, postgrads, and PhDs he has been involved in or completed, but he is also recipient of Young Scientist Award. This was awarded by ISCAI and University of Nottingham, Malaysia, and also got Best Reviewers Award. Today he'll be speaking on transforming waste into value, sustainable recycling of agricultural resources under the carbon peak and carbon neutrality vision. Over to you, Dr. Avasthi.
Thank you. Okay, all right. So good afternoon, everyone. First of all, I would like to thank you, Chairman, for such a nice brief introduction. And thanks a lot. Yeah. No issue, no issue. Yeah, of course, I will be tight on. And then second, thank you very much to Amity University. I'm very delegated to be here to give the presentation in front of all of you. And thanks a lot, Dr. Vivek and all organizing committee members for such a very wonderful event organization in here and inviting us numerous persons across the world to be here and meet together and exchange the ideas to promote the young generation students. So let me start my work and let me first introduce myself. My name is Mukesh Kumar Avasthi and basically I'm Indian. Indian from Jabalpur, is a very small city. And I'm working in College of Natural Resource of Environment in Northwest AF University, China, which is situated, is a very famous place, is Xi'an. You all hear about terracotta warriors. So that place is very close to that And today I'm going to talk about a very overview of my whole work. So talking about transforming waste into value in different kind of the value added product. And modern day, we all, and today conference also, this is sustainability is the most important aspect. Looking the future, especially the carbon peak and carbon neutrality. Most of the governments nowadays is focused on this aspect, how to think about sustainability. And of course, especially the waste valorization. The main point is carbon neutral approach. And in the same aspect, we are working also. Let's here talk about. Okay, let me very briefly introduce my university as well also, just very quick. Our university is situated in Shanxi province, so there is the two provinces, not be confused, Shanxi and Shanxi. Okay, it's just minor differences in spelling. And it's a very exact center of the China. If you put your tips and the center of the China, our university is situated. We have very beautiful two campus as shown in this picture. And recently is completely flourished of the cherry bloom flower. So most welcome all of you if you have a time. And we often organize the conferences, and of course we have many other exchange programs. If you are delegated interested, of course you come here and visit our university and think about and know about our university in China. Let me talk about our university is very famous, and this is the second most agriculture university in China. We are working in the 985 project and also the 2N1 project. These are the two most important projects under that our university. And of course, we have the different other programs is here. As mentioned, there's more than 27 colleges. We have different postdoctoral stations. And there's several discipline area where we are working in the master, PhD, bachelor study. Besides this, we have different other programs. And one of them, as I mentioned to you, that we are the topmost 1% in agriculture sector across the world. All right, now return back to my work. I'm going to very briefly talk about what is the resource. So as I mentioned to here, we all know that waste is everywhere and we human are responsible to produce. None of the other planet where the waste is produced. Even Elon Musk is going to shift us in the Mars, but human is going there, then his waste is there. So the earth is one place where human is generating the waste. And if we want to sustain more long period of this earth, so we have to carefully think about how to recycle our resources, what we are getting back from the earth. Understand? So it's very, very important this point to maintain the carbon neutral approach. All right, we have a lot of the possible technologies as I show here, that biological, chemical, thermal technology. What are the possible way we can utilize them, exploit them, and convert different kind of the value added product? We have a lot of the advantages, disadvantages in different places. There are several regional barriers also restricting us to utilize this technology. Several people don't have those technologies. They're trying to integrate the technologies. That is the possible way to convert and completely utilize and useful production in a cascade process. All right, so we are looking here the sustainability, as I mentioned in the bottom of this picture. So the sustainability is only achieved when you are thinking about the cascade mechanism. Understand? The cascade mechanism integration of technology is very, very vital to convert the waste into several value-added products. And those value-added products can be utilized for several other purposes. All right, these are the resources. So as I mentioned, our university is the agriculture-based university. We have a lot of the resources in here on this picture. We have the several fruits, very fresh. Every day we are eating, enjoying those fruits, especially the apple, orange. We have also several other like blueberry is one of the important foods. And these all is we are considering as a waste because after scrapping out, a lot of the material is a waste. And how do you think about, is it a waste or is it a resource? The human perception actually is responsible whether it's a waste or whether it's a resource. If you change your perception, the waste can be as a resource and utilize use the technology, convert in several valuable product in different forms. This phenomena we took and considering especially the organic waste. So as I show you here, that we have different form of the organic residues, and these all organic residues have different concentration of the starch, lignocellulose, pectin, protein, fat, flavors, and minerals, nutrients. Of course, all the waste, organic waste material they contain. But how easy look like this picture is not that much easy to recycle. A lot of the challenges and selection of the technology is a major barrier to exploit and convert them in a useful product. All right, these challenges we took, as Professor has mentioned, that the challenge is the most important thing you should be accept. And if you accept the challenge, you will succeed and prove yourself. Yes, we have to challenge, but another start the challenge, a lot of the troubles. The first trouble is here. The lignocellulose is a very complex structure. It's not that much easy to convert them in a useful product. We have to look possible way. And as I show you that the challenges is here, that how we can think about the first challenge, the suitable cost-effective approach is very, very important, because we are not doing the academic research only for publication purpose. We have to think about how this technology and can be useful for industrial purpose. That's why we have to carefully watch out from the beginning, and your frame of mind always focused on your challenge and result-oriented work, which can further use for the industry purpose. All right, another point, how we can use the technology. Is it the chemical technology is more useful, or thermal technology is more useful, or the biological technology is more useful? In terms of the science, a lot of the discovery has made in the last 2 or 3 decades, we identified that the biological process is much useful because it's recyclable in several purposes. And then you have to think about how you use these microbes and enzymes. All right. The problems is here, another. The one challenge we not sift and completely finish. The another challenge, this kind of the waste material is also contain a lot of the inhibitory components. I just tell you is for a quick is here, lemon, especially the fruit waste material. And this lemon often inhibit the microbial dynamics, often inhibit the enzyme activity as well also. These 2 points also is a major challenge. Next is here that there are a lot of the inhibitors is present in different kind of the fruit waste material. I listed them, all of them. I'm not going to more explain. Let's talk about what can we do with this material after the pre-treatment, whether we have possible way. Yes, we have met a lot of the research because I work in different organizations. So as I show here in my logo, that one of them university where I'm right now working, and I work in Hong Kong Baptist University, University of Boros, and different other organizations. We work in different aspects to utilize this lignocellulosic waste material to convert into useful product by anaerobic digestion, by the yeast conversion, and of course the fungal cultivations. All of this work is not mine. I'm still too young, so I haven't completed all this work, but it's the teamwork. And my different team supervisors are responsible for this work. I work only the certain area. Those are the I'm going to mention in here. All right, the most important thing here, just a few minutes before, the Mr. Vaskar has talked about the methane conversion, MEC. But I'm not going to methane conversion. I'm going to talk about how to produce the value-added product. All right, the methane and biogas, and of course we know that anaerobic digestion. These 3 important keywords is often connected to use different kind of the organic waste, and of course simply convert to the biogas product. That is the main useful product. It still is very good. I'm not saying that this technology is not good and successful, but a lot of the challenges has this technology due to the selection of the substrate and continuous flow to maintain in terms of the supply chain. So we have to think about if another point that, okay, we are not bothered about the biogas, or we want to be only consider the biogas. And the third question, if the material is not a low biodegradable, what you will do? Yes, we have think about to try to integrate these 2 technology. One of them is AD, and the second one is the versus thermochemical conversion. Both this technology, as you see here, that we have used, and try to be connect how the methane production can enhance by this process. But we identify both process has a certain hurdles. Some of them is not cost effective. The second, another one is that is too expensive. We cannot afford in every place. Everyone cannot afford because of the very expensive instrument, reactors, and maintenance cost is really very not feasible for everyone. All this point we consider, so we consider, okay, let's talk about how do we go for the hybrid methane syngas fermentation. We try to integrate these two technology, considering the biogas production. One of them thermochemical process, we produce some carbon dioxide and try to integrate in our system, and let's try to produce the biogas. This technology, we have MBR reactor we use and try to enhance the biogas production. Try to using the CO2 conversion, MEC just now mentioned, Professor Biogas. But we have identified in this MBR system, there are several safety issues, especially we conduct this work in University of Borås. So we have a lot of the safety issue to damage the reactor with the maintaining the flow of the gases by the thermochemical process. To looking this always, all aspect, we try not to go for this purpose. Understand? We think about another point. Is here, can we produce any other form of the product? Don't bother about the biogas, always AD and biogas. So we try to focus on not biogas. We think about something else. There are several byproducts we can extract from this process. Why we always consider the biogas production? We have several other options, right? So in this point, we have focused, okay, let's talk about the volatile fatty acids. We try to use the MBR reactor and go for the same process, but don't bother about the biogas. We straightforward go for our volatile fatty acid production before the methanogenesis, all right? And this volatile fatty acid, a lot of the industrial demand, so that's the most important thing for us. Okay, let's boost these first three steps. Hydrolysis, aceto and acetogenesis. Don't bother about methanogenesis. We don't need the biogas. But we have the secondary, more clean energy hydrogen by this process, which is more demand for the industrial production purpose. All right, is here. We are continuing extracting by this MBR reactor. And this is a whole experimental setup where we have used the MBR reactor and try to continuous extracting this VFA. As you see this MBR reactor, I'm very happy to inform you this is just a small reactor, but this experiment has almost 7 years before one of our PhD student has done. But recently we have developed very huge reactor by the European grant, is almost more than 1,000 liter, which is industrial purpose. And we successfully launched one company 5 years ago also in Sweden. They are using our technology to continuously extract this VFA product and use this VFA and some other derivative waste material what we after the extraction get out for the animal feeding purpose. All right, so this is the one reactor as I show you that we have the one common reactor in the center of the here, and this is the MBR. In this MBR, we continuously feeding the food slurry, and we have the microbes without disturbing the whole process. We flushing out our main byproduct from the here in this reactor. The time to time, we are also flushing reverse flow to clean our membrane because sometimes the blockage happen. So we try to reverse flow and then clean this reactor, especially the membrane pores. We got some result as you see here that we have applied different organic loading rate, especially 4 organic loading rate. We tested in laboratory condition by using this membrane bioreactor. And we identified that, okay, the higher organic loading rate get out the higher concentration of the VFA. But once we compare about the hydrogen production, we have identified the 8 gram organic loading rate has given us the more hydrogen production, which is relatively very good, understand, in terms of the laboratory experiment. But once we go for the industrial purpose and higher reactor, this production is increased higher amount. All right, these are the products we get out, and we use this product for the several other medical purposes. You can look here, anti-diabetic, anti-obesity product. We are trying to be produce. These are the other possible options. We have to utilize this waste material for several biopolymer productions. I'm skipping out. So this is the most important thing. We are trying to now work in terms of the system microbiology, especially focused on the bioengineering aspect. Look about how strategically we think about nutraceutical approach. The most important thing is here I told you that we don't want to be fluxihilification of our hard work, especially the researchers who are working on this area. So that's why we think about this approach, which can help us to boost the microbial biomechanism to trigger the nutritional properties at the end of the product, and also in the biochemical process, during the process where the substrate and microbes interacting and producing this kind of the biochemical product. All right, finally, I'm going to conclude that once again remind you, the waste is not a waste. Waste is our perception. We have to think to convert or think about first of all that is a resource and convert them in a useful product, not the single product. I once again remind you, don't focus on only biogas. We have several options in this biochemical process. So we think about those area who not touched, especially in the research. Understand? So in this cases, we are chances, the novelty is more higher. And once again, the third one, the carbon neutrality, we have to think about also that how we can utilize and convert in a multi-product, not a single product, which can be useful for the multi-industrial application. All right. And the last but not least, this is the most important thing I personally identify because I'm the very small area, so we don't have the technology. Understand, this is the major drawback for all of young researchers. But if you are eager to work on the right track, a lot of the people come up to you to help you. But your ethics of work is very, very important, and your orientation toward to your work. Just I mentioned, waste is not a waste, your resource. So a lot of the resources surrounding you, but if you are in the right track or right ethics, you can definitely succeed for any kind of the research work. But your orientation should be right. All right, thank you very much. And this is a picture of our group. We are very happy to work on that. Thank you. If you have any question, feel free. You can right now ask me, later on ask me, and you can connect me on the later on by email or by WhatsApp, anything. I'm very happy to be answered.
Thank you. Thank you so much, Dr. Avasthi, for such an interesting lecture. Any questions?
Yeah. Yes.
What kind of membrane are you using?
PEC membrane.
Sorry.
PEC membrane.
All right. One more thing, I saw you have touched a lot of things actually from
Yes.
PEC product. So you have mentioned something anti-diabetic, anti-tumor kind of compounds.
Yes.
So is it tested actually? Is it working?
Yes, we are going, as I mentioned in my presentation as well, we have produced several products by this process. We are testing for the animal feeding purpose.
Okay. Oh, no, I'm asking about the anti-diabetic. I saw one slide.
Yes.
I'm asking whether it is tested actually.
Not tested. We are right now working on this area.
Working on that.
Yes, exactly. We are not confirmed that okay, this product is finally work as an antibiotic. Yes, exactly.
Okay. Maybe a possible product.
Yes.
Okay, thank you.
Exactly. Yeah.
Most of the agriculture industry, or you can say food industry, they are generating the solid waste. And to convert to that solid waste and extract maximum out of it, yes, it can be used after the treatment, it can be used as a feed. But extraction of that valuable product itself is possible with the solid state fermentation, or you'll go for the submerged fermentation and all these things?
We actually solid state fermentation in slurry form, not completely solid state, but
Okay.
slurry form. Try to be identified first different ratio, and then we use for this reactor.
And simultaneously to find out a single product from a group of the fruit food or agriculture waste group. To find out one single product, isn't it? It's a little bit difficult, or
Actually, this organic acid is also itself is a multi-product. There's
Right.
a huge number of the organic
right,
acid.
right.
So we are looking also the particular acid, which one is the higher production. For example, there's a carbonic acid, isobutyric acid. So certain substrate can give you the higher production in the isobutyric acid, but the certain waste material can give you the higher production for the carbonic acid. So we are looking in this area also which particular substrate can give the which particular organic acid higher production.
As we are talking about the as we are talking about the circular economy, the percentage is enough that that cost can be generated from that one.
We have already tested. That's why I told you also that we already launched as a product for the animal feeding purpose, but still we are testing in critical points. So those are the very important concerns you need to be very careful before finding marketable food.
That's true. Thank you.
Sir, I have one question. Just are you able to suppress the methanogen when you block the final things?
We are using some agent. Actually, there is a very easy. You add some agent.
Okay, but BES and other agents are very costly.
Sorry?
Bromoethanosulfonate and other agents are very costly that to suppress methanogen.
Yes.
But still there will be gas, that is hydrogen gas production,
Yes.
that you have shown. But also since this is anaerobic, it will produce little bit of ethanol and other byproduct. So you have to go for distillation. So downstream processing cost will increase.
Yes.
So how to cope up of these things?
We are actually this point very good question actually. We have already for the particular organic acid we are quite successful, and because the membrane bioreactor phases we get the crude form of the fish. Then further go for the distillation purpose. So we already optimized which can give us the minimum cost and high production.
Thanks.
Now this, I had two small questions. And when we do anaerobic digestion, this is 3 or 4 steps where we get the final end product methane. Now, when we use this raw material, agro-waste material, which contains lignin, lignin is recalcitrant. It's a very recalcitrant molecule, aromatic polymer. So, because it inhibits the growth of bacteria. Lignin is bactericidal also. So that one question. So how to deal with that? And second is, since it's anaerobic digestion, and suppose, okay, we will inhibit the methane synthesis, that then we can stop it so that we can get hydrogen and other organic acids. That's a very good idea that you are going. That's fine. But when we, in order to produce the hydrogen and this organic acids here, we have to do anaerobically. Is there any method where we can do aerobically? Yes.
We are testing actually micro ejaculation. Micro aeration. One of our colleagues is also watching in the case.
Sorry to interrupt. Dr. Mukesh, you are waiting for the chair. Maybe we have to survive without tea, isn't it? So it's I think I would do court hall, the main auditorium when we had inauguration. Yeah, thank you so much. Thank you so much, all the question people. Yeah.
So since Sir is again a session chair in I2 Moot Court Hall, we will facilitate him and I'll welcome the session chairs to please felicitate the Sir. नहीं सर वो बॉक्स अभी आ रहा है आपको I2 Moot Court में भिजवाएंगे
So of course we are running a little bit delayed. May I request our next speaker, Dr. Ravi Kant Singh. Sir is acting as a professor, working as a professor and head research, Amity Institute of Biotechnology at Amity University, Uttar Pradesh itself. The research area is in the environmental biotechnology, waste valorization, and he has been leading multiple Government of India funded projects, including MNRE, MSME, MOEFCC, and have more than 100 international publications and patents filed. Currently, sir is involved in UK-supported Waste to Wealth BioGreen project with Nottingham University. With this, I invite you, sir, with your lecture title, Eco-Friendly Production of Biodegradable and Free Water from Bacterial Cells and Waste Resource, a Sustainable Approach. Thank you, sir. Please.
Thank you, sir. Today I am going to talk about the eco-friendly production of biodegradable animal-free leather. I think I already discussed about earlier that we should find out the problem. So we are trying to solve the problem coming from the leather industries. Lot of wastes are coming out from the leather industries. That waste can be converted into the value-added products also. But if we are not creating a problem, making that, removing the problem and by converting it into the eco-friendly leather, so that cannot be create a problem to the society also. So these two aspects we'll discuss about. You all are here in a sustainable biotechnology and bio-innovation conference. So first of all, you should know, ये बढ़ क्यों नहीं रहा? You should know about these sustainable development goals, and my talk fall on which sustainable development goal. You can see that the SDG number 3, good health and well-being, this talk will cover this sustainable goal, as well as we are converting waste into the value, so that you can say that waste clean and waste you can see that this that is not clean and waste sanitation also. I think 2 or 3 sustainable goals are covering out from my research today's discussion. You should know that sustainable development goals should economy or economic also. So that how the circular economy you are understanding. Earlier everybody is talking about the linear economy. When you are converting waste into the value added product, and that created waste, and from that you are giving a economy and making the country more strong, so that this kind of things may comes under reduce, reuse and recycling. By doing this, the circular economy come into the picture. And as in the earlier discussion, sir told that about the waste. If nothing is waste, everything is waste. Depends on the concentration amount, and if you are not able to convert it into the value-added product, that come under the waste material. That waste may be toxic, may not be toxic. If the waste is toxic, you should more cautious about it. But if it's not toxic, then you can find out is it biodegradable, or it may be converted into the value-added products also. So that thing discarded, rejected, abandoned, unwanted, surplus matter come under to the waste. But if that waste is converted into the value by doing the recycle, reuse and these things, you can convert into the certain applications and the biomaterial also. In this slide, you can see that from animal waste, the biomaterials can be produced, and what are the applications are there. Then the fish waste, vegetable waste, rice waste. This means you can target the industrial waste also, you can target the agricultural waste, as well as animal waste and the human wastes also. Each and everything can be converted into the value-added biomaterial. If you are converting into the nanomaterial, it will be more focused and more precise also. And lot of applications are you can see, and you can convert it into your research topic. Today, my purpose is for this discussion is that by listening this discussion, you can make your proposal, make your idea, and under this area you can start your research work also. And this is the purpose of this talk, and it will be deliverable for this discussions. You should understand What is the role of the green chemistry or eco-friendly approach? In my topic, I talk about eco-friendly approach can be done only and only with the help of green chemistry approach, and how this circular economy integrated with the green economy, circular economy, green chemistry and clean tech technology, so that you can protect the environment, you can save the earth and convert waste into the value added product. Now I am targeting about the leather. What kind of problems are coming from the leather industries? These waste can be converted into biogas, earlier discussed, can be converted into energy, in maybe in form of polymers. The packaging materials can be produced, additives, fibers, biochar can also be produced from the waste material, either solid or agricultural waste. But if you are talking about what kind of environmental impacts are there in the leather industries, that leather industries has contained solid waste, has contained liquid waste and the air pollutions also. These solid waste may create some environmental impact, so that I am targeting it, the leather should be produced from eco-friendly aspects also. And what type of the challenges associated with this current leather manufacturing, and how you can overcome this processing processes also, because in the industry the challenges and the future aspects are use of the toxic chromiums are there. So how can you extract chromium from these particular waste, and that chromium can be used from other purposes also. So that you should analyze it also. So that for waste management, it is very much important. First of all, the waste should be characterized. What part is available at the higher extent, so that that can be removed it also. Enormous amount of the solid waste are generating, so that that waste can be converted into the artificial leather, eco-friendly leathers also. It means if we are able to create a eco-friendly leather coming from the waste of the leather industry itself, then it will be the best approach for making this environment eco-friendly. And these things are there. I will cover my presentation on time also. Eco-friendly approaches in the leather prospects are there. The pre-tanning, what kind of things are there, and how we can protect it with the help of enzymatic approach, with the help of drying and other purposes also. Post-tanning, what kind of difficulties and approaches are there, so that eco-friendly approaches may protect the leather processing processes also, and less harmful effect will come to the society. And the waste coming from the leather industries can be converted into the value added product, comes under the waste valorization processes also. That may be liquid, may be solid. If that is water content, then with the help of physical, chemical and the biological treatment processes, the wastewater coming from the leather industries can be in form of sludge, in form of water, and sludge can be used for the biofertilizer purposes also. If you have a solid coming out from that, you can produce biogas, gelatin, secondary leathers, soap and fats, feed and fertilizers can also be produced from the leather industry itself. So that you can say that leather industry is not a problem. You are creating a wealth from the leather industry also, and I am shown this innovative approach also, how this kind of products can be produced from the leather industry waste also. But in this talk, we'll talk about the how can you remove this leather processing industry with the help of the economical approaches, or any kind of agricultural waste, or any kind of waste material you can use for making a leather from this. These are the circular approaches. You can say that from that some other products are can be generated also. Then first of all, you understand what is the traditional leather and what is the eco-friendly leather. What is the difference between these two in terms of the sources, in terms of the production process, environmental impact, sustainability, durability, in each and every, in terms of the cost, each and every aspects, the bio leather, vegan leather, or you can say that the artificial leathers or eco-friendly leathers are far better than the traditional leathers also. As far as the research going on in this particular area, you can see that from 2013 to 2025, continuously leather processing research is also going on, as well as the eco-friendly leather processing also going on. And in 2025 till May, this data is there. Maybe you can say that gradually the level of eco-friendly leather processing processes are increases day by day, and it indicates, it shows what is the importance of this approach and how this approach can be applicable. Because in some case, if you are doing research, we'll discuss about what type of product we develop in our lab. We have patented also this process, and with the help of patent, we protect our innovative ideas, and we can go for the technology transfer also. But if we are not connecting this thing from the industry, so that the large scale production is also very much important. If the production is not coming from at the large scale, then you cannot completely replace the leather industry from the eco-friendly approaches also. So that industry connection is also very much important, and cooperation is also very much important. These are the different approaches: sustainable agricultural waste, what type of products and things can be used. I am going to talk about the bacterial cellulose. And the bacterial cellulose can be grown from the waste coming from the food industry, from the ecological paper industry, cosmetic industry, textile industry, and nanocomposite industries, and what type of things can be converted into it. And I am covering this bacterial cellulose. We are trying to grow this bacterial cellulose in a media, and that giving this kombucha tea. This is tea act as a highly sustainable, biodegradable source for bio leather, offering eco-friendly alternatives to traditional animal leather and plastic-based synthetic alternatives. So if you are using kombucha tree, some other waste materials are also there, but we have patented this aspect from this waste material also only. This is the sustainable approach. If you are able to utilize this tea dust for making this eco-friendly leather, artificial leather, so that you can protect the life of lot of animals also. And this material is cultivated from the symbiotic culture of bacteria and yeast, that is called SCOBY, used to make the drink. If you are using this bacteria and yeast growing onto this tea dust, and that can be used, liquid part can be used as a drink also, creating a cellulose based biofilm that can be harvested, processed, and utilized in the fashion industry as well as in the industrial material, and used as a vegan leather or artificial leather also. As far as this, when you are comparing this, there are a lot of plant celluloses are there. So why you are targeting this bacterial cellulose only? So that bacterial cellulose have a high purity, nanofibers, high crystallinity, excellent mechanical strength, and high biodegradable capability. This comparison already being done, and due to this, we can conclude it that bacterial cellulose will be the better thing for converting it into the sustainable animal alternative to the animal leather and all. If you are talking about the sustainability and environmental impact, why this thing should be there in terms of the biodegradability, the first important thing, low energy and zero waste is also there. Waste is converting into the value, and low toxic chemicals in coming out when you are creating a vegan leather in a industry. So that these are the processes, how to the fermentation process. In place of sugar, we have used molasses. And in terms of kombucha tree, we use tea dust. So that the waste and molasses is also waste, growing this bacterial cellulose and making this agricultural artificial leather, and that leather can be used in some other industries and cosmetic industries also. This is the control aspect, and this is the test sample done in our lab. Here the molasses replaced by the sugar, and tea leaves replaced by the tea dust and all. These are the process for making this thing.
kombucha as a sustainable source of metabolics, the fermentation processes are there, what type of alternatives are there, and where we can use this. This can be used for the economical packaging materials also, because this is the biggest challenge nowadays. In a packaging material, the packaging materials are also non-degradable. So if you are able to use this leather in a packaging material also, that can be easily degradable and protect the environment in that particular aspect also. This production microbial aspect and what type of yeast you can use, which bacteria, and lactic acid and bacterial these compositions, and the then characteristics and the application of the production in terms of that, because if you are going to produce anything, the characteristics is also very much important. Is it practically feasible? You created leather, but from that leather, have you developed anything and all? If you are able to make the mobile cover, and from this leather also, that can also protect the environment in smaller aspect also. So I will show this. These are the summary analysis, the microbial growth we analyze when he compared with this, and why this is better in all aspect. These things are given on, and what is the cost. You can see that when you are using the control unoptimized media, 7 rupees, and that is in around 2 rupees. It may be reduced also when further researches are may be gone, will be done. As far as this diagram I can see, so this thing, these the figures came from our laboratory, where we developed this artificial or vegan leather in a lab from the kombucha tree, and that is being utilized for using as a pen holder, as a key thing, and mobile cover also. So that in smaller aspects you can say that, but if we'll work with the industry, because we file a patent also, and after that we'll go for the technology transfer and in connecting to the industry, then industry will help you also with, because we are the academician, we are small researchers. If the industry peoples come, then that knowledge, laboratory experiments will go to the field also. And when the lab experiments go to the field, then that field work will help to the society also. So that's we are trying to connect with the industries. These are the future prospects I already covered due to time scarcity, and I suggested for the I am trying to suggest Government of India also for promoting this eco-friendly leather also. Why this eco-friendly leather not coming into there? The first and important thing is infrastructure and policy support should also be there from the Government of India. R&D and innovation funding should also be there, as well as the market promotion and awareness. If these three things will not be there, then any kind of research will dump into the laboratory only, that will not go to the field, that will not go to the society. So that thing is there, the green industry revolution, and then from my side, anybody can ask anything.
Thank you, Dr. Ravikant. Anyone has any question for sir?
Sir, thank you for such a wonderful and very important presentation, sir. Sir, in leather, the collagen is so strong because of amide bond, peptide bond.
Yeah.
Here you are using cellulose,
Yeah.
which is a beta 1,4-galactoside. So I am just asking, do you think that this is mechanically that much reinforced?
Yeah, it's checked also. The mechanical strength and durability is also been checked in the laboratory, because the things should not be more stable. That flexibility is also important. As
Yes.
far as civil engineering is concerned, if you are making a bridge and that bridge is very rigid, that will be break.
Yes.
That bridge should be flexible also. The flexibility is the strength, not a weakness.
Okay. Yes. Of
Sometimes, but if we are making a formulation and all, and that is mechanical, having the mechanical strength also.
Yeah.
That's why
not only cellulose,
Yeah.
the other
Other one. That's why we converted into the mobile covering and all, and this is the that will be flexible, it not be possible.
Sir,
Mobile.
I have another question. The tea contains lot of polyphenolic compounds.
That will be
Do you
removed
think
from it.
that it has
Then
some
the
role?
characteristics is required. If that initially I told that if you are using waste
First of all, the waste characterization is important.
Yes.
What is essential, what is not essential. You cannot directly use any waste as a layman. First of all, we characterize and which is important. If chromium is not important, first of all, extract chromium. And if any silica is creating a problem, you remove silica. And then go ahead according to.
Thank you, sir. Thank you.
Thank you. That's why the chemistry people may also help in the
I was also interested in that data of TGA, DSC, and young modulus, like how strong, how whether it is ductile, brittle, or how it behaves actually compared to leather. So once we'll have T session, we'll discuss.
I'm sure Dr. Ravikant must have done all his testing for the characteristic because he has patented.
patent also.
So if he has patented, that means first let that come, right? Yeah. Okay. Thank you, Dr. Ravikant. I think with this we conclude the session. Thank you.
Yes, ma'am. Now we invite our session chairs to felicitate our invited speakers. I would like to invite. अच्छा मुकेश सिर्फ रविकांत से बचे हैं हाँ वही बचे हैं हेलो रविकांत से इनवाइट और कहाँ से बात फोटो खिंचवाएं रविकांत सर Thank you, ma'am. Thank you, ma'am. I would like the session chairs to please stay back. And Ravikant sir, aap bhi please for felicitation. Ravikant sir, aap bhi. I would now like to felicitate our session chairs. Ravikant sir, Rajni ma'am, Subodha ma'am. हाँ जी नहीं आइए बेगम yes अरुण गोयल सर I would also like to invite you to felicitate session chair, sir. Please, please, sir. May we have Thank you so much, sir. Thank you so much, ma'am. Uh-huh. Thank you so much, ma'am. Thank you so much, sir. Thank you so much. It was a wonderful session and we truly enjoyed it. I think so the students also enjoyed the session. We'll move to the section now of the evening and क्या sir sir का felicitation. I invite all guests now to please come back on the stage and do the honors. Thank you. So, ma'am, आप भी तो चल उनका बॉक्स यही तो दोनों ही तो बोला मैंने 2 डब्बे Thank you so much. Thank you so much. We are starting with our next session.
भी अफोर्ड कर सकते नहीं ना
ये नए सेशन अपने गेस्ट को बुलाओ उनको कम से कम ले जाना चाहिए ना हमारे नए वाले को लगाओ और वो इनका वो कहाँ है आगे लगेंगे इनका नाम क्या है सेशन रविकांत सर प्लीज कम ऑन द स्टार्ट कर हाँ जी सर थैंक यू सो मच सर
Good evening, everyone here. On behalf of MIT University, Noida, organizing committee of International Conference on Sustainable Biotechnology and Bio-innovation, SBB 2026, it gives us immense pleasure to welcome you all to the session of Environmental Biotechnology. We extend a very warm welcome to our esteemed chairs, Dr. Ravikant Singh and Dr. Rajni Singh, whose contribution to microbial enzymes in biotechnology contribute to inspire research and innovation. We also warmly acknowledge our session in charge, Dr. Gurumurthy, for his guidance and support in shaping this session. A special welcome to our distinguished invited plenary speakers, respected faculty members, researchers, industry experts, and our enthusiastic students joining us today. Your presence truly enriches this platform for acknowledged exchange and collaboration. This conference, organized by MIT University, Uttar Pradesh, in association with the Biotech Research Society of India, International Bioprocessing Association, and the Center for Energy, Environment and Sustainability, with Unipic and IUth as a gifting partners, stands as a testament to our shared commitment toward sustainable biotechnology and bio-innovations. We look forward to insightful discussions, meaningful interactions, and inspiring ideas throughout this session.
Today's program is designed to be both engaging and impactful, featuring insightful keynote lectures, thought-provoking presentations, and interactive discussions. We encourage all presenters keep their talks clear, focused, and time-bound to ensure an enriching experience for everyone present. With that, let's begin today's proceedings. And now I have the honor of introducing our session chairs, Dr. Ravikant ji and Dr. Rajni Singh ji. Dr. Ravikant Singh, sir, I would just say sir, sorry, is working as professor and head at Amity Institute of Biotechnology, Amity University, Uttar Pradesh, Noida. He did his MTech Biotechnology from Institute of Engineering and Technology, Lucknow, and PhD in Biotechnology from IIT Roorkee. He has completed various research and consultancy projects in the field of environmental biotechnology funded by Government of India. He has published more than 100 research papers of international reputes and patent filed on waste valorization, environmental sustainability, biofuel and bioenergy. He has completed various research projects funded by Government of India like MNRE, MSME, MOEF-CC and CG Biodiversity Board in the field of environmental biotechnology. He is also working on a bio-green project under research team on waste to wealth, sponsored by Nottingham University, London, UK. We welcome you, sir. May I have a round of applause for our first session chair? Moving on to our next session chair, Dr. Rajni Singh ji. Dr. Rajni Singh ji is a distinguished microbiologist, academic leader, and innovator who has made significant contributions to environmental and industrial biotechnology. Yeah. She is additional director at the Amity Institute of Microbial Technology, Amity University, Uttar Pradesh. She holds a PhD in microbiology from G.V. Pant University of Agriculture and Technology, Pantnagar, and has over 2 decades of experience in teaching, research, and academic leadership. With over 2 decades of experience in research and higher education, Dr. Singh has established herself as a pioneering women scientist in the areas of microbial technology, nanobiotechnology, waste valorization, and sustainable enzyme technologies. She has published more than 100 research papers in high impact international journals, holds 24 patents, authored books, and contributed extensively to scientific literature through book chapters and invited articles. Her research on microbial enzymes, nanomaterials, biodegradation of industrial pollutants, antimicrobial resistance, and agro-environmental sustainability has generated both impact as scientific and societal relevance. Dr. Singh has successfully led and completed multiple externally funded projects supported by national agencies such as DST, DRDO, ICAR, and the Ministry of Environment and Forest and Climate Change. Through her visionary leadership, commitment to mentoring young scientists, especially women researchers, and dedication to sustainable science, Dr. Rajni Singh ji exemplifies excellence in women-led scientific innovation and academic leadership. We welcome you, ma'am, over here. And with that, we welcome the session chairs to kindly take their place, the chairs, and we start with the session. Thank you, ma'am, and thank you, sir.
Thank you, sir.
Thank you. Now I will request Dr. Santosh Kumar Mishra for a invited talk. He is a professor and head, Department of Life Science, Sarda Universities. He has more than 21 years of teaching, research, and academic experience. He did PhD from IIT Roorkee, and I was fortunate. I supervised him as a supervisor also. And he had an experience of industrial biotechnology, bioprocess engineering, downstream processing, and his PhD work is also related to the enzyme. Did all the experimental work at IIT Roorkee, but I requested my supervisor to kindly supervise my first student under your supervision, and she accepted. And due to this acceptance, he also did the experiment under my supervision also. Thank you, and we achieved a lot of things in terms of academics, in terms of industries exposures, and I'm proud to see the level of Dr. Santosh Mishra here and going to present his talk in this important topic, challenges and opportunities in sustainable production microbial enzyme using cost-effective agro-industrial waste as a substrate. Over to you, sir.
Okay, thank you, sir. I am very thankful to my guru, Professor Rajnikant Singh, and I am also thankful to organizer of this conference, Dr. Amit and Dr. Vivek for inviting me. And from morning we are listening lots of lectures, delivering sustainable biomanufacturing and other things. But personally, I believe that in nature there are 4 orders broadly, that is material order, plant order, bio order, and human order. And role of 3 order is definite, but due to knowledge, human growth is continuous. But now human has created lots of problems for rest 3 orders, and we are setting the sustainable goals and many more things. But once we realize that if we are committed towards saving and preserving rest 3 orders, there will be no problem in nature and surroundings. Okay, so my message is to young audience: whatever we are doing in research, whatever doing in our day-to-day life, please think about rest 3 order also, so that we can live in this in a sustainable manner. Okay, so now I am starting my discussion, that is challenges and opportunities in sustainable production of microbial enzyme using cost-effective agro-industrial waste as a substrate. Enzymes have gained great interest worldwide due to their wide variety of biomedical, scientific, and industrial applications. Improper disposal methods such as landfill decay or open burning significantly contributed to environmental pollution and global warming, especially in terms of agricultural waste. Annually, a large amount of waste is generated by the agriculture and food sectors. Some of this waste is used as a soil fertilizer or animal feed, while some needs to be treated to avoid environmental problems. And India generates an estimated 686 million tons of crop residues annually, with Uttar Pradesh leading in generation of 60 million tons, while in Europe up to 37 million tons of agro-industrial waste is produced from the food and beverages industries. This situation becomes critical if we consider that agro-industrial waste increases with an increase in human population, which is estimated to increase by 9.3 million people by 2050. There is a great demand for a consolidated process that critically evaluates the current stage of knowledge, providing insight into multiple enzymes export under SSF conditions, employing various types of low-cost lignocellulosic biomass, including agricultural food waste. Here is a data, and in this slide we will see that from year to year that agricultural waste is generated is increasing significantly in year-wise, and this data is relatively low. But in this data, we will see that in the year 2017 and 18, there is a constant rate of increasing crop residues versus biomass buried in India. So this is a problem in somehow, but there is a solution in here, because agricultural waste can serve as a low-cost solid material for the production of industrially important compound, whether it may be organic acid, biofuels, or microbial enzymes. So we have a lots of potential. Since India is a agricultural dependent country, we are significantly growing in the area of AI, computer science, and many other related areas. But most of the population at present also dependent on the agriculture. So there is if a agriculture field is growing, their agriculture waste is also growing, and by using this waste, we can generate a value-added product. There is an opportunity for a country like India and other developing countries. And food waste, if in terms of food waste in India, up to 40% of the food produce is wasted. For example, 21 million tons of wheat are wasted in India. According to the Ministry of Agriculture, INR 50,000 crores worth of food produced is wasted annually, and USA produced 1.3 billion tons of waste a year, which worth an estimated $165 billion. And overall data in terms of cereals, fiber crops, sugarcane, oilseed, pulses, and others is mentioned here. And based on the work I have carried out from more than 10 to 15 hours in the microbial enzyme production, recently I published a review paper that titled Microbial Enzyme Production Unlocking the Potential of Agriculture and Food Waste Through Solid State Fermentation. So most of the things I have summarized in this review paper. So here in this slide, we will see that chemical composition of some agriculture food waste that allow for the production of industrially important enzymes. So here we will see that wheat bran, cassava peel, and so many other things in terms of content-wise mentioned here. So in this data, we will see that carbohydrate, protein, and lignin is the most common compound that is present in the agricultural food waste, and these agricultural food waste content must be utilized for the production of low-cost value-added products. And here in this slide, we will see that the fermentative microbial enzyme production process. So agricultural food materials like a wheat bran, cassava peel, coffee waste, sugarcane waste, and so many other agricultural waste can be utilized through a set of fermentative process for the production of value-added products. So in this slide, we will see that agricultural waste can be utilized both way in the way of solid state fermentation as well as solvent fermentation for the production of value-added product like the microbial enzymes. Okay. And here in this slide, we will see that agricultural waste produced country-wise, and this is a very extensive data. We have researched a lot of research paper to summarize this data. So in this image, we will see that the agricultural waste produced by different countries. So here we will see that US that is producing nearly 25% agricultural waste, India producing 21% agricultural waste, and China producing significantly low agricultural waste, and after that Brazil and some other small countries is given here. But here, if in terms of microbial enzyme production, the companies or industries involved in the microbial enzyme production, you can see that in the USA, specialty enzymes and probiotics, enzyme solution, Singapore, DuPont Industries, Davidoff International, and Fortis Industries are the major industries that is producing the microbial enzyme using low-cost agricultural waste. And in this area, India is also leading because there are industries who are constantly using agricultural waste for the production of microbial enzyme. So here are the few industries, that is Advanced Enzyme, Advanced Biotechnology Limited, Biocon, Novozymes South Asia Private Limited, Rosari Biotech Limited, Kemin, that is South Asia Private Limited, and in the China, the major enzyme producers is Beijing, Science and Technology Company Limited, Ribomonas Bioproducts China Company Limited, and in the Brazil, Biocell, Phytobios, Genpor, and Insulin is the major industries involved for the production of microbial enzyme fermentation process. And you may be surprised that these industries are using low-cost industrial waste for the production of microbial enzymes. So here we will see that there is a lot of potential in the agricultural food waste as well as other agricultural residues for the production of microbial enzymes. And this is the summary of schematic downstreaming of steps involved in the production of commercially viable enzymes like xylanase, cellulase, alpha-amylase, pectinase, lipase, etc. using agricultural food waste as the raw materials. So this is the summary. So you can see it, and this paper is available online. You can see it. So here I want to summarize some industry important enzymes that have a very commercial importance, like xylanase, pectinase, cellulase, lactase, lipase, fructosyl transferase, amyloglucosidase, and alpha-amylase. Out of these, I have completed my research work in the area of alpha-amylase production by using biomastic indicators under the supervision of Singh sir and Professor Suresh Kumar sir, which is my mentor. They have guided me, and they have created a lot of interest for doing this work. Initially, I was interested to do my research work in the area of plant biotechnology, but sir has insisted that this area is having lot of potential. Therefore, I have done lots of work in that area, and thank you, sir, for guiding me. So here, different types of microorganisms along with enzymes is summarized here that is involved for the production of microbial enzymes. So, and in this slide, we will see that the advantages of agriculture waste in microbial enzyme production. So enzymes are of great importance in the industry due to their substrate and product specificities, moderate reaction conditions, minimal byproduct formation, and high yield. They are important ingredient in several products and production processes. Up to 30% of total production cost of enzyme is attributed to the raw material cost. So if we are using agricultural waste as a raw material, definitely this will reduce the overall production cost of enzyme. The food industry expels obvious amount of processing waste annually, which is mostly lignocellulosic in nature. This can be used as a raw material with a set of biochemical reaction. for the microbial enzyme production. Upon proper treatment, lignocellulose can replace conventional carbon sources in media preparation for industrial microbial process such as enzyme production. However, wild strain of microorganisms that produce industrially important enzymes show low yield and cannot thrive on artificial substrate. So there are different types of effort has been made to mutate or modify the microbial enzyme for better production of industrially important enzyme. And at the same time, recombinant DNA technology is also playing a very pivotal role for improving the different types of strain for better production and better yield of the enzymes. And here are the challenges in using agriculture based in large scale production of enzyme. One major challenge is substrate heterogeneity and pretreatment, because pretreatment process increases the cost of enzyme. And in the agricultural based lignin, hemicellulose and cellulosic materials and their complex network creates a lot of problem in the use of agriculture based as raw materials. And there is a challenges in the scale of because microbial nature of biochemical processing nature of different type of microorganism is different. They need different types of physicochemical conditions for the growth. There is a major challenge for scale up. And another is economic and infrastructural challenges. Country like India have not developed sufficiently for economic and infrastructural challenges that we are facing for using the agriculture based as a raw material for the production of enzymes. And there is a technical and environmental obstacles because government is not providing sufficient technical and environmental challenges that the researchers are facing. So that is the major hurdle in the use of agriculture based at large scale for the production of enzyme. Here is the potential solution and trend. Research is focusing on using genetic engineering for strains that can utilize complex ways, optimizing SSF for larger scales, and using pre-processing method to minimize the creation of inhibiting compounds. And this is the current research we are doing in our laboratories. Our few students is involved in the production and process optimization. So this is the summary of work that one of my PhD student is carried out. And another group of students is working in the area of silicate production. Only few researchers is at present working in these enzymes. So I am also working in this enzyme because this enzyme is having ability to degrade the silica containing compound, especially the silica which is present in the rice straw and other agriculture waste. This enzyme can degrade the silica containing compounds. And here we may surprise that silica containing compound like silicic acid can be used as a but it can be used as a compound that increase the fertility of soils. So we have isolated different strain of silica solubilizing bacteria, and this is the method and characterization. And here we will see that silicon oxygen group is present, and this shows the presence of silicic acid by FTIR. And this is the optimization strategies for the silicate enzyme production. And here another student who is presently working as isolation and optimization of alkaline proteins producing bacteria from the soil. So one of Nigerian students who is working under me, he is presently working on that enzymes. And this is the basic methodology and screening. And these are the optimized conditions for protein production. And this is our research group. Here, this is a student, international student, that is Ali Jabel. He is working on the enzyme protease. And another is Yamini. He is also working with another my PhD student, Oinam Vidyarani Devi. They are jointly working on the enzyme silicase. And Deepali from the right hand side, she is working on the linkage enzyme production. And I'm thankful to Sarda University for giving this opportunity to present my work. Thank you so much.
Very good lecture, Dr. Santosh Kumar. It's a really good opportunity to see that the waste material can be used for the production of the microbial enzymes. Any question? Now the session is open for the questions. If anybody has any questions? I have only one question. I have only one question. As the silicase point, it's a really big issue in our northern area that because of the burning of the rice straw is a major issue. So does the amount is enough that we can treat the field or for the treatment of the field?
We are treating the silicate nitrogen from different locations.
Okay.
To isolate a effective microorganism that will produce a sufficient amount of silicate, and at the same time, if microbial culture along with several other culture, so we are
Consortium.
We are
Like consortium.
Consortium.
Okay.
For degradation of rice husk, because the rice husk is a major problem in the northern area.
And the degradation is because of the presence of the silicate itself.
We are working on a harmonistic approach for degradation of agriculture.
Good. Thank you. Thank you, Dr. Santosh.
Nice presentation, Dr. Santosh. And he covered the problem of silica because in agricultural, in the rice husk, silica creates a lot of problem. Sometime in a biogas generation also, silica decreases the rate of biogas production also. Right now, government and everybody is talking about the bio-CNG that contain biogas also, and a lot of opportunities are there for doing research in that particular area. Thank you, Dr. Santosh, for giving some light on it. Thank you.
Our next speaker is Professor Arun Goel, please. He is from the Indian Institute of Technology, Guwahati. So,
Make the full screen here itself, also not in the windows.
he's from the Indian Institute of Guwahati, and he's a prominent scientist in the area of enzymes. And publication wise, he's having more than 300 publications in his kitty with the fine H index. He supervised 38 PhD students and the PI of more than 20 projects. Among them, 3 were international. He has been awarded with the Distinguished Scientist Award 2024, BHU Continent Award 2020, Malviya Memorial Award 2016, and Excellence in Carbohydrate Research 2017. He is a fellow of different societies, including the AMI, BRSC, NABM, NAS, and IFIB. Over to you, Dr. Goel.
If the slides are in, I want to say one thing. This conferences is sponsored by BRSI also, Biotech Research Society of India, and students must register and being a member of this BRSI. Lot of opportunities will there when you attend conferences funded by the BRSI. Okay. Over to you, sir.
Actually, मैं शेयर भी करना था एक बार अगर इससे हो जाए तो
Yes. A very good evening to you all. And first of all, I heartily thank the organizers for inviting me here to give and present my work here, what we do in the lab. So today I'm going to present a very small problem which we have solved, say the new enzyme, which is a postgraduate work in our lab. And my previous speaker also mentioned about one word, which was recombinant DNA technology. So this is the use of that technology which we have, by which we have created a new enzyme, cloned a new enzyme and expressed it and fully characterized it. So this is slightly more into deep into the basics. So I hope audience have some background in biochemistry. So I'm going to deal with a little bit of enzyme chemistry and also what this enzyme does. And we know that each enzyme is very unique and each enzyme has a very specific role to play, a specific reaction to carry out. And each enzyme is specific for a particular bond it cleaves or it creates. So this is the title of talk is a processive exo-1,6 galactonase from Ruminococcus flavifaciens. What is this is plant cell wall and pectin. Pectin is the most complex polysaccharide. Just focus on this, most complex. And the constituent of this is the constituent of a plant cell wall, and which is found abundantly in the primary cell wall or and in middle lamella. And pectin is made up of, just see, 17 distinct monosaccharides with over 20 different linkages. So this is one of the most, we're going to see the structure as well. It consists of homogalactouranin, xylogalactouranin, and apoyogalactouranin, and also known as smooth region of pectin. And pectin also contains highly branched hairy region known as rhamnogalactouranin, which is in short RG1 and RG2. RG1 side chain are rich in galactans. So I'm just narrowing down from this complex structure to the galactans and coming to my enzyme, which we have cloned and what the activity of that enzyme is. So RG1 side chain are rich in galactans and also arabinogalactans, which serve as substrates for galactan degrading enzymes. So here you can see the structure. Middle lamella is shown, primary cell wall, and you can see the pectin, how it is in terms of like strings, it is connected or it is binding to or it is keeping together cellulose and hemicellulose content of the cell wall. Then the right hand side shows the layout of homogalactouranin. You can see this in short, and then XG, xylogalactouranin, and apoyo, and then RG1 and RG2. So the next slide is going to be clear. So there are two types of galactans, type 1 and type 2. So type 1 contains beta 1,4 linkage and type 2, 1,3 and 1,6. This is how they have been characterized or classified. So galactan type 1 is beta 1,4 and occurs as a side chain in RG1, rhamnogalactouranin 1 pectin. And galactan type 2, which contains 1,3 and 1,6 linkages between galactose side chains, or it could be side chain and also it could be main chain. So this is highly branched region, and other substituted, this can also be substituted with side chain of arabinose residues. And this is found in RG1 and arabinogalactan proteins AGPs. And these are again very structurally complex and recalcitrant. Now just see this structure here. There are different enzymes by which different mode of action of these enzymes can degrade the galactan type 2. So if you can see this here, these are galactose residues linked, and this is like an endo 1,6 galactonase. So this is cleaving this bond which has 1,6 linkage. Likewise, there could be exo. Now we need to understand the exo and endo concept. Endo enzyme will cut any polymer or any compound from within the molecule, and exo is going to cleave only from the side. If this is a large chain, so exo enzyme will act only from the end and will remove stepwise 1 or 2 monosaccharides. So that will be exo. So today's enzyme is, our enzyme is exo 1,6 galactonase, you see. Now this targets 1,6 glycosidic linkages in type 2 galactans, and even if the galactose is substituted with the side chain of arabinose. And it cleaves from the non-reducing end, not inside, so end, releasing small sugar units like galactose or galactobiose. So we are calling it as, that's why it is an exoenzyme. So let's see. And we know the application of galactonase. It has all those applications which are also covered by cellulases or xylenases. So it is applicable to all like paper industries, food, beverage, drug, biomass, valorization, animal feed, and even production of galacto-oligosaccharides. Excuse me for my throat. Now, the organism which we have selected is Ruminococcus flavifaciens. So, what are the characteristics of this organism? This is a gram-negative anaerobic bacterium found in the rumen. That's why it takes this ruminococcus. So, rumen of cattle and also hindgut of monogastric animals. It breaks down cellulose and plant cell walls using specialized multi-enzyme complex called cellulosomes. That's why we have taken this, cellulosomes. Cellulosomes, they contain a large repertoire of complex enzymes which are involved in degradation of carbohydrate enzymes. And these cellulosomes play a crucial role in digestion and nutrient cycling in the ruminant animals. Its genome has a variety of carbohydrate enzymes. That's why we have taken this now, making it a model organism for studying human polysaccharides degradation.
Slide is not.
Oh my God, you should have told me. Yeah, let's go. Okay. Yeah. So applications I mentioned, that this takes all those applications covered by cellulase and xylenases. And the Ruminococcus flavifaciens, why we've taken? Because this is anaerobic and it is found in rumen, and it contains this cellulosome which has multiple enzymes, and its genome, it was found with several carbohydrate enzymes, which makes it a model organism for studying these human polysaccharide degradation. This I've already explained. So why galactonase? In this genome, what we found that a family GH30 was not characterized biochemically, and so its exploration could reveal some new catalytic features and with new substrate specificity, which could be relevant for industrial applications. And what industrial applications? Say biomass conversion and also production of these oligosaccharides, which can serve as prebiotics for nutraceutical industry. Now, this is the enzyme which we have focused here. Now, this shows the molecular architecture of RFGH30, because this belongs to the glycoside hydrolase family 30, and small f refers to the full length gene. So, this architecture shows from 1 to 860, the full length gene of the GH30 enzyme. So, first 30 amino acid residues shows the signal peptides, which is followed by a catalytic module GH30, which is a catalytic module, followed by a carbohydrate binding module family 62, and then also a dockerin, because dockerin will always be there because this is a cellulosomal enzyme. So now the gene encoding, what in this study, what we focused, the green part, from 31 to 505 residues we cloned and expressed. So this is the catalytic module of the whole gene was taken and cloned. And this was expressed. I'm not going to the molecular biology aspects, which was cloning to this PET vector, and this was expressed. You can see also the activity staining in orange by Congo red, and it is active enzyme and also was counter stained. So this confirmed also the expression and single protein, no contamination, single band protein was expressed. So successful and soluble expression of the protein. And below shows the purification table from the crude to the IMAC purification. If you know the immobilized metal ion affinity chromatography, which we use the nickel ion chromatography. So this purification was also achieved, which is now pure single band, single protein. And this enzyme activity was obtained. If you see units per milligram was 6.6 through this purification table. And this was taken after purification, this was taken further for its characterization, because this is a new baby which we have got. It was not pre-existing. So we need to characterize, and also we need to really find out whether this is the same enzyme which we anticipated for. For that, we have to do for assay. So first of all, we need to check because this is a mesophilic organism. So the optimum temperature was 40 degrees and optimum pH was 5.5 in the acidic range. This is thermostability. It showed above 66% enzyme activity in the temperature range from 30 to 60 degrees. pH stabilities in a wide range from 5.5 to 7.5 pH. It could retain 90% activity, and the half-life was also determined. So half-life was 145 hours at 4 degrees and 14 hours at 30 degrees. And the last D plot shows the melting temperature, how it can sustain the high temperature. So TM analyzed by Boltzmann fitting was found to be 50 degrees. So these are the characteristics or parameters which we need to determine so that we can take it further for any other application. Now comes this is new baby, so we need to really name the enzyme by checking the specific or this activity against various types of substrates. So in the top, we can see this it's active against arabinogalactan, which is from larchwood source, and this company name was Magazyme. Because we need to purchase these substrates from different companies, and they have different type of purity level. So we always mention also the name of the company so that we can remember which company is giving better results. So like Sigma, we also have, so there is always some variation in the activity. So in the top, after optimization of the pH, temperature, everything, these activities were determined, and the maximum was with arabinogalactan. So that means you have a galactan chain and arabinose is the side chain for an around 8 units per milligram. And next substrate was arabinon, followed by galactan, followed by rhamnogalacturonine. And then there was some little activity with also xylene substrate, arabinoxylene, very, very low activity. And in the bottom, if you see, these are the synthetic substrate also we've checked. So against PNPG's para-nitrophenyl beta-galactopyranoside or beta-galactoside, which is giving 5.4. I'll come to that later. Now, this enzyme exhibited RFGH30 without the F small, exhibited preference for type 2 galactan-rich polysaccharides, so which are generally arabinogalactan. This enzyme showed no activity against beta-lactose, which we checked, is showed in the table also the last beta-lactose from Sigma. So that means this does not have, so we have to we rule out that this does not have beta-galactosidase activity. Beta-galactosidase is, which will cleave the galactobiose. Only the biose is degraded by this enzyme, beta-galactosidase. So this enzyme does not have this activity. It showed significant activity with synthetic substrates. Now, this confirmed if this is giving very high activity. If you see 5.4 with the synthetic substrate, that means it is demonstrating exolytic. So it is cleaving because this ONPG is a very small molecule and it is able to degrade this and release this nitrophenol. So that means it can act on the 1 or 2 residues of a large chain. So this is also, this confirms that this has exoactivity, not an endo so far. So from here, we got some substrate against which it is acting, and also another information we got that it is an exoenzyme. Now again, since we have optimized all those conditions, so we extended this study to other substrates which were showing significant activity like larchwood, arabinogalactan, arabinon, ONPG, PNPG. So this is the kinetics VM, Vmax, Km, Kcat with all these substrates. So exogalactanase, now we have started naming. If you see, we are now calling it as exogalactanase. Again, we have not specified 1,3, 1,4, 1,6. We have to go further. So far what we confirmed, exogalactanase, RFGH30 exhibited maximum affinity and catalytic efficiency against larchwood arabinogalactan. This is just routine stuff. We have to check also if it is a metal enzyme. So we check against different metal ions, the effect of these metal ions against this activity. So we can see the calcium is able to raise by 20%. So it is, yes, it is metal dependent enzyme, and calcium is one of the best so far in this study. So it can also enhance the kinetics. So it can raise the it can activate the enzyme for towards higher activity. So exogalactonase, this enzyme was activated by calcium ions and also to some extent by magnesium, because calcium and magnesium both fall in the same family in chemistry. Now coming further analysis, mode of hydrolysis by this exogalactonase by TLC analysis. So we have taken again largewood arabinogalactan as a substrate and did the time analysis with time. The cursor not showing here. So anyway, if you see the TLC chromatogram, you can see the minutes 1, 5, 30 minutes, then 1, 6, 12, and 24 hours. And these are the standards. S1 is arabinose, S2 is galactose, and S3 is galactobiose. Now, see here, 1, 6 linked. Now, there could be a possibility of bios having 1, 3 linkage, 1, 4 linkage, and 1, 6. And all will migrate with different RF factor or different distances on the TLC. So we have chosen 1, 6. We had anticipated that this is going to be 1, 6. So we included that substrate bios here. With time, we can see that there is always 1, 6 galactobiose formation with time until 24 hours, starting from 1 minute. So we need to also check the initial rate kinetics, initial, what is the product, just to detect whether it is purely exo or it also has endo, or endo activity got converted to exo later on. So since 1 minute also the same product, until 24 hours also the same product. And also there is a followed by some formation of galactose on the top until all this. So mainly we are getting majorly two types of product, galactobiose and galactose. So 1, 6 galactobiose as a major product and galactose as a minor product were released. And this is analysis of the same tubes on the right hand side. We can determine the TRS by estimating the reducing sugar. And that goes also like this with the after say 8 hours or so, or maybe in first 4 hours, the formation of reducing sugar is saturated and it stays constant up to 24 hours. Now, so this confirmed also that this is releasing, or this is the enzyme is acting on beta 1,6 linkages. Okay. Now in this experiment, in this TLC, we have taken galactose as the standard here, and then galactobiose again as a control here, and then also just the galactobiose 1,6 left it for the same time duration along with the enzyme. So 2 minutes. Yeah, just 1 or 2 slides here. Just some explanation right here. Sorry. So, and the last lane is with the enzyme. So we can see there is no formation of galactose from here, from galactobiose. And some autolysis also taking place when we are not using enzyme with time. So this again rules out that this is not a galactosidase enzyme. This is only exo. There is no formation. It is not acting on the galactobiose, 1,6 galactobiose. This is again routine HPLC analysis. This confirms the same TLC results. These results corroborate the same TLC results, the formation of only biose and galactose. These profiles show the same. This is the standard and this is arabinogalactan. You can see the same 2 peaks, galactose and galactobiose. And with another substrate also sugar beet arabinon, same 2 substrates. So again, this shows, now we can say this is beta 1,6 exogalactonase. This is just again the same thing. This is from mass analysis again, at what time, which are the moieties which are from. So we found again galactose and galactobiose. We need to confirm from all directions, from here, yes, these are the 2 products. Otherwise, these publications, they never believe us. So HRMS. Now, this is again processivity index. It showed very high processivity index of 20 with the largewood arabino. Processive means it methodically cleaves one after the other the last galactose in a fashion manner. So this is a processive enzyme. So this is purely exo, and this is also a processive enzyme. So this is just a conclusion, but what we have done so far. I've already explained everything. So this enzyme, I'll just read the last line. This enzyme underscores the biotechnological potential of selective galactobase production from any biomass we can extend it to. So this is the gist of the whole study. So I thank you all for listening. This is the publication coming out from this work in carbohydrate research. And this is my group. Thank you for listening. Sorry for taking over time.
Thank you, sir, for an excellent talk, and you cover a lot of data into it, and maybe the students will be benefited for this. We are very happy to see you, a professor from IIT Guwahati, delivering a talk, but very few students are there. It needs more than 100 students should be there in this. Okay, no problem. But we required, I think, if can you accommodate, because in our NTCC students in final year, the students went for the dissertation works. PG and the PA undergraduate students. If you allow our PG students, MTech or MSc students for doing dissertation at your laboratory, and it will be beneficial for our students also.
Every now and then we get these requests, and we also take many students during summer. Now. So that's
Yes. Great for us.
For dissertation, it becomes difficult for longer period because of the scarcity of our hostel facilities. But summer it's always available.
Summer is also okay.
Already.
Some students also for doing the summer internship. Thank you, sir. Thank you for giving talk. Anyone ask anything?
Any question? Yes.
So, selective, what was the expression level? What was it parallel to the indigenous one, the native one, or the
Was it much increased? More than 20% of the cell protein was this assigned. Otherwise, we will not take it further.
Okay. And the expression vector was designed like it was modified designed, or it was commercial one? So which one was used? PET 20. PET series only you could get it.
Widely used.
Thank you, sir.
Great. Thank you.
Recorded format. Recorded format. Recorded लाइव नहीं मैडम का करा दो रिकॉर्डेड बाद भी चला देना हाँ रिकॉर्डेड बाद भी चला देना तो उसके बाद चलाऊंगा सो नाम करो तो 9 दिन का हो ही गया ना टाइमिंग आपका हो गया आपका क्या बजे 6:10 का था 6 तो आपका टाइम हो चुका है आप अपना करिए रिकॉर्डेड बाद में चला देंगे बाद में चलो है ना इसके बाद ही चल रहा
जैसे सेशन चेयर्स मांगेंगे
if recorded I think it will be better than recorded if the person is online then it will be difficult possible तो ये लगाएंगे ना उसके बाद
Fine. Good evening to one and all. I'm Dr. Smriti Srivastava. I work in capacity as Associate Professor here at Amity Institute of Biotechnology.
Sorry, I will introduce you.
It's absolutely fine.
Yeah. Students, Dr. Smriti Srivastava, she is a senior faculty at Amity Institute of Biotechnology, actively involved in the research works, and she has a laboratory in the ground floor enzyme technology lab, and did PhD from BITS Pilani or BITS Misra, having more than 15 years of experience. And she completed a lot of projects and published good research papers also. And she is also taking care innovation and entrepreneurship activities at the university level. And she is an active member of IIC, Ministry of Education at Amity University, Noida. Over to you, ma'am, for delivering a talk on microbial enzyme-based valorization of biodegradable
अच्छा ये अलग से लगाया
waste. Thank you.
Fine. Thank you. Thank you so much for the introduction, sir. And so I'll move ahead. The topic which I'm presenting today is microbial enzyme based valorization. So it all started right from my PhD work when I started working with xylanases. From the microbial sources, and the target for that those xylanases were application in paper and pulp industry, and slowly we could move ahead with various aspects of it. So what we can in general see here, the valorization of agriculture and municipal waste is something which I have with my baby steps in the research work with whatever funds available. I have been able to come towards it. And we see there are numerous opportunities when valorization is has to be taken care, and obviously we have to pick up few of them where we can ideally work, which for my work area it's actually works with production of some specialized molecules or biochemicals and enzyme production in total. This is something which I am working in my lab with the help of one of the PhD students. She has already done with one PhD, she is working, and then there are many internal research students from BTech, MSc, etc. All the programs, and they work together. So the initial idea, what as I said, was it all started with xylanases and then came to the applications and the xylanases and clubbing them together for a better efficiency of the same. So I'll start with the glycoside hydrolases initially, what we work, and we all understand, all of us, in today's discussion, we have seen this numerous times. So what are the challenges? The majorly dealing with the hemicelluloses one, the challenges being with the lignins. So we ideally have to make a club of enzymes together in order to get it. So in this process, and then the first project was for the value-added products production, which I'll come later. Now, as the distinguished delegates earlier have also mentioned, and sir has mentioned that before working on any particular thing, we should be very clear about the pathways, how it works on. So this is something for the one step into the bioethanol where we understand, and the saccharification process. So let me tell you, my major work is on increasing the efficiency of the saccharification of the municipal as well as the agricultural waste, which I have now included along with the sugars. I have also included the proteins with my second PhD scholar, wherein it has come. So this saccharification with the xylanases can be briefly explained by this. And this is the primary work for the xylanases from two fungal species. Those were both identified as aspergillus only, but they could produce good amount of xylanases, and in consortia with another xylanase from this source, I would just say, yeah, from this particular source, that is Thermomyces lanuginosus. So we have ideally studied the entire aspect right from the screening, then getting the proteins, then purifying the protein from Thermomyces to homogeneity. And when this aspect was studied for aspergillus, it was not purified to homogeneity, but it was partially purified, and the crude one was analyzed with various in cocktails in various combinations to see the saccharification efficiency, and we could ideally get an enhanced saccharification efficiency as compared to any other reports available. So that and with all these, we have already filed patents which are in process. These are the complete patents filed, and we have research publications also from this. So what you see over here is the protein profile. What you see over here is the effect of the metal ions. Then obviously this picture enhances that, it tells you about the TLC. This picture, this here also, what you see over here is a TLC picture, and this is from the strain that is Thermomyces lanuginosus, which was worked priorly. And this TLCs can be very well seen that what you see here after the effect of the enzyme is only the monosaccharide one, which this particular property of this enzyme actually adds a lot to the cocktail, because it is very readily converting all the oligosaccharides into the single units. So this was added here. This is a layout of yeah.
So once we have already seen the cocktail effect of these enzymes, specifically xylanases, then the next approach was bringing them together in the nano carrier or the nano composite forms. And for the nano carrier forms, we have already made the nano carriers, nano composites, and we have, this is also the process of filing the patent is ongoing, but then it is already approved. The basic study is already done. I do not have the number. That's why I have not added the file number as of now. Once we got the enzyme, so the primary step of saccharification was done. So saccharification is what? Getting the sugars. After getting the sugars, it needs to be valorized. So in order to valorize it, I have not moved too much into valorization, but valorization has been done with few of the things like xylitol, ethanol and all. So for ethanol, what we have done is like ethanol production through the native strains. We also have now ethanol production normally happens with the 6 carbon sugars, right? Saccharomyces if I take. So for that, we have also requested few strains from universities abroad, which were the recombinant strains earlier as well, which could ferment the pentose sugar, and as well as we have also constructed few of the strains here, modified the Saccharomyces for a consolidated bioprocess system. But the consolidated bioprocess system, as in bioethanol, we understand that the conversion cannot be more than 52% because of the formula itself. And among 50, in that 52 also, if you take the native one only with the single step fermentation process, the efficiency is higher. But with the consolidated system, which we could design in the prior project, we could not attain the efficiency, but the working was attained, wherein the consolidated system was developed using the cloning and the respective expression vectors. And we could see here, you could see here the things. This was also analyzed and through the process. The production, the cocktail over here was then, this is the production unit from the pre-treated. And then since I said that it majorly works with the agricultural residues. So whatever here, this is being worked with the corn cobs, sugarcane bagasse and corn cobs were majorly being studied. So when we study those, all the aspects of the untreated, treated, how the changes in the morphology and all, everything has to be studied. So these are few of the pictures which speaks about that. Then obviously FTIR complements that, because you can understand the change in the functional group using that particular thing. So all these were clubbed, and then the effect on based on the of the pre-treated ones on the ethanol production was analyzed. Ethanol production with one of our senior faculties, Dr. Monica Prakash Rai. I'm sorry, I missed to write ma'am's name over here. She was earlier with us. Now she is with MNIT. And ma'am worked with on biodiesel production. So when you work on a biodiesel from the algal sources, you actually we defat that. So that defatted ones, the sugars ones were used, and we worked for the ethanol production. We have the final processing of this is actually ongoing, and we are up for a publication of this particular work. Another problem with ethanol production, major problem with the industry faces, is the problem of sugar tolerance and the tolerance of high toxicity due to the ethanol itself. So we have collaborated this. We have MOU with one of the industries here, that is Catalyst Biotechnologies, and we have collaborated. We have got some of the samples from the company, and then for the target given by the company was, like, we were actually planning for a combined project with the company. That is the high tolerance strain, the strain which could tolerate high. So with the students, these Aditya and Ashiv, I think I've mentioned the names of all the students. This is Dr. Meenal, who has worked, Malika and Manaswi, then Aditya, and they were working on this particular aspect, and we have been able to get some mutated strains based on various approaches which we could work on, and those strains are able to produce ethanol at a relatively higher sugar tolerance, sugar concentration, which we can see. But then this is also at the initial stage of the work. Moving forward, like apart from the cellulases is something which we have never studied in our lab, because a number of them are known. Even xylanases number are known, but number of them are known. But because of the xylanases from Thermomyces lanuginosus, which was very specific, we took this xylanases forward and the production. Then we also understand sir was telling about pectin and polygalacturonase, which can be also clubbed when we are speaking about municipal waste specifically. So we we have this set of organisms which are working, and we have analyzed it for this cocktail of these enzymes, which helps in the production of the which helps in the hydrolysis of pectin as the source. And this we have also, yeah, the patent for this is also filed, and the basic research, one of them has been published, and the other ones we are coming forward with the applications one to be published. Next target is obviously the proteases that has been worked by Miss Shalini Chaudhary. She is a PhD student, and she has started with the isolation and screening of the protease producing organisms from various sources. And we have, yes, got few significant isolates, we can say, which can work on various aspects. Although Shalini's PhD target is not with the valorization of the waste material, but she has some other objectives for her project. But here we are also picking a few for the combined making for the waste valorization process itself, wherein we can see here some alkali and the acid tolerant proteases have been identified. Few pictures are the graphs are some shows the effect which we have seen on the hydrolysis from the proteases, which we are working in our laboratory. So what I actually wanted to convey is that we are in the process, and I'm actually in the process of moving towards the cocktail of these enzymes of whichever and all the aspects, whichever steps we have taken with all the set of enzymes we have already seen, and we have already patented all the processes there. And then with this, we will move ahead with further applications, whether be in the valorization, or we have also identified, students have also identified few other applications wherein we will move ahead with that. And so few of the applications which I've currently covered is these particular things. And yes, I had all the works from the beginning have been yet supported by the funding agencies, right from the CSIR research fellowship, wherein we get the contingency, and then followed by 2. But then yes, I'm looking forward. Actually, change of. I'm looking forward to some help from funding agencies for taking this forward, because without a good fund, a good criteria in the works is
Nice presentation, Dr. Smriti.
Thank you so much, sir.
And I want to know one thing.
Yes, sir.
When you are dealing with enzyme during for the degradation of waste, okay,
Yes, sir.
so what type of challenges you are facing in a laboratory?
So when pre-treatment was done earlier, when with the xylanases, when mostly extensively xylanases I have studied with the students and myself. So the major challenge was the conversion, and that conversion because of the lignin. So then we have already standardized. We have published that also in the Journal of Bio-FPR journal. It was published, wherein we standardized various protocols for delignification. And then when with the delignification approach, when I spoke to some other faculties, then the whole idea of making the nano carriers with the lignin ideally came. So delignification helps a lot, sir. So if we have good delignification approach.
Are you using the immobilization process also for that? Because the enzymes are very sensitive.
Composites I'm using, but not the basic immobilization one. Composites.
It works.
Yes, sir. It is working.
Okay, thank you. Thank you.
Just cellulose is also there, because cellulose-free xylanase is a little bit difficult to find it out.
So, ma'am, the one from Thermomyces was cellulose-free xylanase. But the one from Aspergillus, the two from Aspergillus, they were not cellulase-free xylanase actually. They were clubbed together, and even they were not purified. But because of the T-land one, it works better. And the other cellulase which has been taken, that we have not taken from the lab.
So in a composite treatment, have you used both of them? Cellulase-free and also, because a cocktail type of thing, if you have used.
For the Aspergillus one, when it was used, it was with the cellulose one, with the cellulose one.
Yeah,
please. किसी को पूछना? Okay.
Within the organism that cellulose was present, cellulose or
No, normally xylanase doesn't come cellulose free. Matlab, it is always a combination with that. But then we were lucky, lucky in the sense ki earlier when I studied with that, just 1 second. When I studied when we were studying with the TLAN xylanase, and that TLAN was completely purified and then studied, the sequence and all was studied. That was an added point there. So that could help. Otherwise, it is very difficult. And for aspergillus, when we studied with aspergillus one, then we moved towards a different track. It was actually not purifying that, but then making a consortia, making a combination cocktail of that, and then clubbing that with the other enzymes. So there we could attain an enhanced saccharification. So that was not interfering. And we also use some enzymes from the company. So those were mentioned in the protocol.
so earlier from first organism where you are saying you got only xylanase. So you made that from a cocktail using that enzyme also from external cellulases, or you just use it as in xylanase?
When if only purified xylan is there, then that xylan from the thermomyces will obviously work better. But when we were working on a sample that is raw, there we cannot just put that xylan. So that has to be combined with the other things. Now there were various combinations, either the commercial ones which has already been reported from the companies. We also collaborate with companies. We also get enzymes from the company. We also compare that with the company's products. So how they are working. So that is how it comes. So if it is a raw material, and if it is a pure xylan, then their efficiency was very high. I could not actually explain it. It could saccharify, it could hydrolyze that within 12 to 15 minutes. So that we have reported almost 10 years back. So it's in process. Yeah.
Thank you.
Thank you so much, sir.
Next lecture.
Our next speaker, Dr. Raj Bhupati. He's from the Nicholas University, USA, and he's a distinguished scientist. Have more than 250 publications, more than 14,000 citations. That is the major thing which he is having, and he's working on the bioremediation, wastewater treatment, and bioethanol production. His talk is on the presence of antibiotic resistance bacteria and antibiotic resistant genes in recreational water of Southeast Louisiana, USA. Please.
This was done by my grad student, the organizers for the invitation to present. 100% of this work was carried out by CESC conference. This work was done by my grad student, Taylor Andrzak. This first of all was done by my grad student, Taylor Andrzak. Bacteria in the water exist. This first of all was done by student bacteria in the water. If you look at bacteria in the water, they exist in two forms. They exist in the form of planktonic, like free living, or in the form of biofilm. They attach to any substrates and they grow. One of the common substrate nowadays you find in the waterways are plastic. So bacteria attach to this stick and they form a biofilm, and the biofilm mature and become bigger and bigger, and then it slough off and disperse and go find another substrate. And nowadays you have so many plastic bottles in the water body, so there is a lot of opportunities for the bacteria to attach and grow and have a big issues in aquatic ecosystem. So we set up a biofilm control plastic bottles in recreational area and did our research and to show the difference between free-floating bacteria and biofilm bacteria, particularly the antibiotic resistance. So how antibiotic resistance develop? Antibiotics end up in the environment from various sources. Could be from pharmaceutical industries, hospitals, from agriculture and food, sewage disposal. When it ends up in the environment, there are the biodiversity of bacteria in the environment. So there are a lot of germs around, and few naturally have drug-resistant properties. And when you have these antibiotics in the environment, it kills the antibiotic-sensitive bacteria, and as a result, you have only more antibiotic-resistant bacteria will start to multiply. And once they start multiply, they start exchange genes, and we call that horizontal gene transfer. As a result, the antibiotic resistance spreads very quickly in the environment. And this is one of the pathways of why antibiotic resistance is spread very fast in ecosystem. So the horizontal gene transfer occurs by three different processes. First one is conjugation, where gene transfer happens between two live cells. You have antibiotic resistant and bacteria that donates piece of the genes to recipient bacteria, and the recipient bacteria, once it acquires the new gene, now it becomes antibiotic resistant. And then another way antibiotic resistance spread by horizontal gene transfer is by transformation. In this case, when bacteria die, especially when antibiotic resistant bacteria, the genes are exposed to the environment. We call that free DNA. The free DNA, when it comes in contact with a cell that are competent, they have a right receptors in cell membrane. They acquire the free DNA and transport inside and insert this free DNA into the genome. Now this bacteria become antibiotic resistant with a new property. And then transduction happened through accidental gene transfer via viral replication. When virus replicate, especially the bacteriophage, which attacks only bacteria, sometimes it accidentally transfer bacterial genes into new bacteria and then transfer antibiotic resistant to the new bacteria. These are the three ways the horizontal gene transfer occurs. Once the bacteria acquire antibiotic resistant, then genes spread very quickly in the environment through vertical gene transfer, which is every time they reproduce, and bacteria reproduce in mostly logarithmic scale, and it spreads very, very quickly. Let's look at how antibiotic works. There are two kinds of effect of antibiotic on bacteria. One is called bactericidal, which results in death of bacteria. Another is bacteriostatic. It prevents the growth and replication of bacteria. Example of bactericidal is antibiotic that disrupt cell wall synthesis. The bacteriostatic antibiotics are that can prevent protein synthesis, replication of DNA, RNA in bacteria. Antibiotic resistant mechanism happen because bacteria develop resistant genes for various ways these antibiotic work. Some of them are they develop alternative process to avoid using the antibiotic target. They also inactivate the antibiotic by secreting enzyme that can break down antibiotics. They limit the uptake of antibiotic by modifying the protein pump present in this cell membrane. They also get rid of antibiotic through efflux pump. As soon as the antibiotic gets into the cell, they pump it out. And also they modify the target of the antibiotics, so antibiotics cannot go and bind it in the target. So these are some of the ways bacteria develop resistance to antibiotics. So if you look at the history of antibiotic, and it's a major global issue nowadays. So before 1920, when there was no antibiotics, we had a lot of infectious disease that was a major way that people get killed. And once the antibiotic was discovered in the early 20s and become commercially available in mid-30s during Second World War, a lot of lives have been saved because that infectious disease spread was limited. And that's a period from 1930s, mid-30s to 70s. We call this golden era of antibiotics. A lot of different antibiotics were discovered. There are 20 plus new classes marketed. And first resistance developed as soon as the wide prolific use of antibiotic was used, resistance were developed. And the MRSA, which is the methicillin-resistant Staph aureus, which was first noticed in the 1970s. And from 1970s until now, there is a big void in antibiotic discovery. The pharmaceutical industry moved away from producing antibiotic to go to make money-making drugs such as lifestyle disease like obesity and other, problem people have. So there are less research in new discovery of new antibiotics. And this is leading to a big issue of by the year 2050, annual deaths from antimicrobial resistant bacteria expected to exceed cancer-related deaths, which is kind of scary. So we're going back in time. So we're going to have more infectious disease going to spread. So coming back to antibiotic resistance in the U.S., in the U.S., you can see the map here. This shows the antibiotic prescription by 1,000 people in a population. And Louisiana is number 4. Out of 50 states. They over-prescribe antibiotics, which is misusing antibiotics. This is one of the causes for antibiotic resistant development. If you look at West Virginia, there are 994 prescriptions per 1,000 people. And a population. Louisiana is called Sportsman's Paradise because we have a lot of recreational activity in the state, and we have a lot of fishing and hunting. If you look at the license issued for fishing, so we looked at 2 sites. The first one is called Ataka Pass Landing Site, which is located in this map. And you can see this boat launch vessel. The Pleasure Bend site also has a lot of activity for fishing. And if you look at these both sites side by side, you can see the land use pattern. So here's the Atapaka's landing for boat launch. A lot of agriculture and some residential fishing camp is red. So our main objective is to monitor water quality in these 2 boat launches and isolate pure culture of bacteria, determine antibiotic resistant and susceptibility and antibiotic resistant genes present in the water body, and also monitor bacterial diversity in both places, both in free form and also in biofilm. So specifically, we looked at water temperature, pH, dissolved oxygen, chemical agent demand for organic carbon, specific conductivity, nitrate, ammonia, sulfate, and phosphate. And for pure culture of bacteria, we studied various enteric bacteria like E. coli, Enterobacter, Klebsiella, Enterococcus, and Staphylococcus. And we looked at various commonly used antibiotic and one rarely used antibiotic, which is meropenem. And then other antibiotic that commonly prescribed from amoxicillin to all the way to penicillin, we monitored in the water sample. And then we also looked at the biodiversity of bacteria, both in free form and also in planktonic form, and both sides. And look at the succession of bacteria from, what's happening during So to quickly go through the method, we had a water sample collected from both sides, triplicate for 1 year. Every month we collected. We used the HAC colorimetric method for water quality analysis for chemical parameters. We used fecal and total coliform analysis using MPN method, most probable number method. And then we isolated pure cultures and just a little bit more details about chemical analysis. We did this HAC spectrophotometer method, very easy to do. And these are the different methods we use for various analysis for nitrate, ammonia, sulfate, phosphate, and organic carbon, which is use chemical oxygen demand. We use this spectrophotometer DR6000 for this analysis. And then for total coliform, fecal coliform bacteria, we use the MPN method. Total coliform samples are incubated at 35. Then biodiversity. Then we did the antibiotic disk assay using Kirby Power chart and used these various antibiotic disk and concentration of the antibiotic used given here. And you can see the if there is a no zone at all, bacteria grow close to the disk. That means that bacteria is resistant to antibiotic. If it is big zone, the antibiotic is working and the bacteria is sensitive to this antibiotic. Quickly look at the result. When you look at the nitrate concentration, both in pressure band, which is the blue line, and the Tacopos landing site. If you look at ammonia, ammonia also has the same trend. No significant difference within the two. If you look at sulfate, sulfate told a different story. We see a clear difference in sulfate concentration in the Tacopos landing site. There's high sulfate. Then pH, pH is slightly alkaline in Tacopos landing site compared to pressure band. There is a statistical difference between these two sites. And you can see the p-value. And also within the season, we saw. If you look at antibiotic content, this is the antibiotic concentration in the water in nanogram per liter. We took sample in triplicate in the fall and in the summer. Here's the average of different antibiotic in the water. We never detected meropenem, which is good. So meropenem is the last resort antibiotic, very rarely prescribed. But we found all the antibiotic commonly used in Louisiana in the water bodies, amoxicillin to tetracycline. Specifically, we find high concentration of sulfamethoxazole, trimethoprim, and tetracycline. And these are statistically different compared. If we look at total coliform bacteria, and there is statistically we find pressure band. And if you look at fecal coliform, it tells the same story. And we found one sample site has, and during pressure band, had up more fecal coliform counts and fecal coliform count. And then my student Taylor did a lot of isolation of pure cultures, and you can see she isolated 207 of them for antibiotic resistance. And then we did the phylogenetic analysis, and you can see if you look at the diversity, and in terms of multi-drug resistant, we found isolate that are resistant to 3 or more antibiotic of 3 different families of antibiotic. And if you look at the resistance in terms of isolate and convert them to percent of bacteria that are resistant to isolate, resistant to antibiotic, you can see the R is resistant. And we looked for the presence of antibiotic resistant genes, and we found antibiotic resistant gene commonly we found in the SAL2 gene for sulfonamide resistance. And we looked at the antibiotic resistant gene for basic tracing antibiotic. For ampicillin, same story. Every isolate we tested that are. So to summarize the chemical parameter, fecal coliform abundance, Tacopos landing we find antibiotic content. So why sulfamethoxazole, trimethoprim, tetracycline are highest? Because of use. If you look at agriculture, especially, we can see this agriculture is main activity. So cattle farming is one of those. So all this water. So what it means we have to take care of the ecosystem. We have to follow this One Health. So that's the recommendations listed here. We want to continue to monitor these sites and increase sampling frequency. We want to include research in other, boat launch facility, other recreational area. We also want to do source tracking using human papillomavirus. Management-wise, we want to prevent antibiotic use or reduce antibiotic in the environment, fix septic systems that are not working in the rural areas, and reduce plastic pollution, which is important because the biofilm shows that rich in bacterial diversity and rich in antibiotic resistance. This work was again done by McGrath and Taylor and Razak. The work was funded by Koipu Foundation. And I would like to stop here and hopefully you can send me.
Stock students, you can do research in this MDR, I think, multi-drug resistance system, and it's very good research you can do in future also. If any kind of help and suggestions or any kind of guidance required, you can send a mail to this professor, because we are not able to ask any question in this moment. Thank you.
Thank you, sir. Yes, sir.
Not mentioned, sir.
Sir, mail ID has not been mentioned.
Big or may provide उन्होंने invitation तो भेजा ही होगा ना, and that video came from that particular email. So that email share with you all people. Okay, thank you.
Should we conclude now? Thank you, sir. And this brings us to the conclusion of this session on microbial enzymes in biotechnology. Today, discussions have highlighted innovative approaches and critical perspectives towards sustainable biotechnology and environmental solutions. With this, we come to this end of this session. And to begin with the felicitations, I would like to invite Ravikant sir and to please be Dr. Sir. Can you run you? Yes. Thank you so much. Thank you so much, sir. Thank you so much, ma'am. I would now like to Shraji. Can we have the round of applause, please? देख लो चेंज करवा दो वो बाद में चेंज कर देना वो बैठ जाए ना तब चेंज कर दो Last but not the least, Dr. Srivastava. Can we have a round of applause, please?
part of yoga.
Thank you, ma'am. Thank you, sir. Thank you. Nahi, please, the section chairs, please wait, sir. Hanji, 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 Hanji. Now, since I'm the senior most faculty over here, I would like to have the opportunity to felicitate Ravikant sir and Ranjit ma'am. Kindly give the gifts, please. And our session in charge, Dr. Please come. Last but not the least, before we conclude, I would like to express my sincere gratitude to esteemed chairs, Dr. Ravikant sir and Dr. Rajni Singh ma'am, for their valuable guidance and leadership throughout this session. I also extend heartfelt thanks to our invited and privileged speakers for sharing their expertise and to all faculty members, researchers, and students for your active participation and insightful interaction. My special thanks to the organizing committee of SDB 26, Amity University of Uttar Pradesh, Noida, and a collaborative partners BSI, IBA, Biotechnology, and CESS for making this session a huge success. We would also like to extend our heartfelt thanks to our gifting partners and IOT. Thank you all for your time. And we conclude with the session. Thank you so much. And I invite all the delegates for the dinner. Thank you so much. There's a small change. I